उसी की स्पीड से आदमी वेट भी उठाता फटाफट में। अच्छा बॉडी उस समय तो गर्म होती है, जब बॉडी ठंडी होती ना तो नगाड़े वाले गाना अगाड़ा पिछवाड़ा सब है उससे नगाड़े जैसा हो जाता है। उसके बाद जब बॉडी ठंडी होती है फिर आदमी ऐसे उतरता है।
तूने ये जो नगाड़े के साथ अगाड़ा और पिछाड़ा लगाया ना, ये बहुत तगड़ा था।
अच्छा इन्हीं गानों पे हो सकता है, आप देखो दूसरे कभी गाने पे हो सकता है। चंदा है आदमी कर ही नहीं सकता, चंदा है वो डम्बल साइड पे रख के आदमी सो जाएगा कि छोड़ो परा। अच्छा ज्यादातर लड़कियां जब जाती है ना स्पेशली जिम में, वो मेकअप करके जाती हैं, पूरा ऐसे लिपस्टिक पूरा कट कर जिम में ट्रेडमिल में भागते भागते भी तो देखती रहती हैं। नहीं लड़कों का ध्यान इन पे होता है, लड़का इधर भी देख के एक्सरसाइज कर रहा ना तो उसका ध्यान पीछे ट्रेडमिल पे भागती लड़की पे होता है। लड़की अपना मेकअप करके ना सीधे ऐसे जो भी करती रहती है, फिर 1 महीने बाद कहती मेरे वेट तो कम हुआ ही नहीं। अबे मैडम मुंह धो के आओ, 2 किलो मेकअप उतरेगा फिर देखना। सॉरी गर्ल्स बट होता है ऐसे। 5 मिनट के वर्कआउट में 15 फोटो खींच लेती है, और जो ट्रेनर इनको जिम कराने के लिए आया था उसको फोटोग्राफ मत।
अरे यार ये गो टू मीटिंग में जा रहा हूँ तो ये तो कुछ और ही खुल रहा है। ये इसमें आ रहा है मीट नाउ शेड्यूल मीट माई मीटिंग में जाऊँ।
हाँ तो वो माई मीटिंग में तो जा।
हाँ माई मीटिंग में गया, ठीक है। अब यहाँ आ रहा है एकेडमिक सेक्शन एमबीबीएस फर्स्ट। दो एमबीबीएस आ रहे हैं, ऊपर भी आ रहा है एक और दो नीचे भी आ रहा है।
अरे 20 वाले में मत जा, 20 वाले में जा, 20 वाले जो दूसरा है।
कितनी बार, 2 बार क्लिक करूँ?
हाँ 2 बार
कर दिया। वेलकम टू गो टू मीटिंग। हाँ भाई ठीक। अरे महाराज इसमें वो कहाँ है लॉक अनलॉक वाला? लॉक अनलॉक वाला कहाँ है? वीडियो कॉल कर रहा हूँ मैं, वीडियो कॉल। हेलो 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 मैं सुशील हूँ। वो अभी है नहीं यहाँ पे।
अच्छा वो नहीं, तो आज आप कर रहे हो?
हाँ जी।
ओके। आपने ऑन कर लिया?
हाँ मैंने ऑन कर दिया, स्क्रीन शेयर कर दिया। ऑफिस से लेंगे ना क्लास आप?
मैं नहीं ले रही हूँ। माइक ऑफ है ना?
माइक ऑफ करा हुआ है अभी,
ओके हाँ ये
कैमरा
चीज़
भी
मुझे,
ऑफ करा हुआ है।
ये मैं कल मनमोहन जी ये ऑन छोड़ दिया था।
अच्छा
ये मैं भी चेक कर, इसको मिनिमाइज नहीं करो।
किसको मैम?
ये जो है ना,
समझा नहीं, मतलब किसी को मिनिमाइज ना करें, इसको
हाँ
इसको
गो
कह
टू
रहा
ऐप, नहीं नहीं नहीं, इसको खोलो। अच्छा मीटिंग स्टार्ट नहीं हुई है?
नहीं, मिनिमाइज हम नहीं करते मैम, वो मीटिंग स्टार्ट होगी तो अपना आ जाएगा ऑटोमेटिकली, शेयर करना पड़ेगा, प्रेजेंटर बनाना पड़ेगा।
अच्छा अभी स्क्रीन वो स्टार्ट नहीं की है
नहीं
शेयरिंग।
अभी
ये मनमोहन जी ऑन छोड़ देते थे और कल थोड़ी दिक्कत हो गई थी इससे।
माइक से?
हाँ जी माइक
नहीं
ऑन,
माइक तो ये रहा मैम, ये ऊपर ही है, इसको ये ऑन
तो
करना है, इसको
हमने
ऑफ
ध्यान
करना।
नहीं दिया ना।
अच्छा
हमने ध्यान
कौन से
नहीं,
डिपार्टमेंट से मैम आप?
क्या डिपार्टमेंट ऑफ एनाटॉमी से हूँ।
अनॉटमी से हैं आप?
वही तो
अच्छा
कल दिक्कत हुई थी बहुत, आज भी दिक्कत हुई। तो कुछ इंस्ट्रक्शंस दिया, बोला है उन्होंने मनमोहन जी ने। आज राजीव सर आए थे ना?
हाँ हाँ।
आप थे उसमें?
कौन से सबडिन
हाँ
राजीव?
हाँ जी हाँ जी
हाँ जी हाँ जी।
थे ना? सर ने क्या बोला आपको?
नहीं मैं नहीं था मैम उस मीटिंग में। अगर आपको इन केस अगर कोई प्रॉब्लम होती है ना क्लास में, कब है आपकी क्लास?
मेरी क्लास नहीं, मैं अभी हमारी डिपार्टमेंट्स के मैम की क्लास है एक।
अनॉटमी की?
हाँ तो मैं अटेंडेंस लेती हूँ यूजी का, तो
अच्छा
इसीलिए मुझे, तो उसमें बहुत अनवांटेड बाहर के लोग आ गए थे
उस ऐप
क्लास के अंदर। हाँ और फिर कुछ कुछ पोस्ट किया था।
अच्छा वो तो खैर मैम हम भी कुछ नहीं कर सकते उसमें, लेकिन हाँ कुछ ये बोला है कि लॉक अनलॉक कुछ बोला है करने के लिए, तो
बट
वो
वही है
मैं
ना,
कर दूँगा।
लॉक तो आप कर दोगे, ठीक
हम्म
है, बाय चांस वो बच्चे का नेट चला जाता है तो क्लास से बाहर हो गया ना वो,
हाँ,
तो वो फिर रीजॉइन करेगा। अब लॉक हो गया ये क्लास, तो रीजॉइन कैसे करेगा वो?
वो तो करना ही पड़ेगा हाँ।
वही चीज़ मैं जानना चाह रही थी राजीव सर ने क्या बोला था, मैं थी नहीं सुबह में।
नहीं नहीं सर ने तो सिर्फ ये कहा है कि आप
लॉक कर दो।
लॉक कर दो।
ये आप रिकॉर्ड नहीं कर रहे हो ना अभी?
रिकॉर्ड भी हो रहा है।
स्टॉप कर दो रिकॉर्डिंग, आप जब क्लास स्टार्ट होगी
नहीं नहीं
तभी
मतलब अभी हमारी रिकॉर्डिंग नहीं हो रही है।
गो टू ऐप की जो अपनी है वो रिकॉर्डिंग हो रही है।
हाँ जो अभी आपकी जो देखो माइक ऑफ करा हुआ है ना,
हम्म।
हमारी रिकॉर्डिंग नहीं हो रही है।
ये जो आपने खोला ऐप, गो टू ऐप, ठीक है, ये लाइव स्ट्रीम भी करते हैं ना एलटी में, तो ये लाइव स्ट्रीमिंग हो रही है या नहीं हो रही है?
लाइव स्क्रीनिंग नहीं हो रही है अभी, क्योंकि लाइव स्क्रीनिंग तो मना कर रखी है ना, बंद करा
अच्छा
हुआ है ना मैंने।
बंद कर दिया, कौन सा लाइव स्ट्रीमिंग वाला?
ये बोला,
नहीं
कैमरा
भैया,
बंद कर दिया ना मैंने।
आप नहीं समझ रहे हो। LT2 जो यूट्यूब है
हाँ
उसमें भी लाइव स्ट्रीमिंग होती है,
हाँ
मैं वो पूछ रही हूँ उसके रिगार्डिंग। ये जब क्लास स्टार्ट होगी ना तो LT2 सिम साइमल्टेनियसली कोई भी बच्चा LT2 यूट्यूब में जाके देख सकता है ना? दैट
क्या
मीन्स
क्या क्या चीज़ देख सकता है मैम?
क्लास।
एक्चुअली मैम हमें भी ना नया ही दिया है सॉफ्टवेयर ना, तो अभी हमें भी थोड़ा थोड़ा इतना नॉलेज नहीं है, और दूसरा अब जो आप कह रहे हो वो मेरे को समझ में भी नहीं आ रहा कि वो यूट्यूब में जाके लाइव देख सकता है।
यस हाँ तो वही मैं जाना चाहूँ, वो होता कैसे हो?
कौन से मैम के मैम क्या नाम है उनका?
डॉक्टर अरुंधति शर्मा, उनकी क्लास है अभी।
अच्छा ठीक है, एक मिनट रुकिए, फिर मैं देख लूँ, उनको स्क्रीन शेयर करनी पड़ेगी ना, एक मिनट।
वो आ गई है क्या, पहले आप चेक कर लो।
अरुंधति बोला ना?
हाँ डॉक्टर अरुंधति शर्मा उनकी
यही
क्लास
है
है।
ना,
हाँ डॉक्टर
अरुंधति हैं खाली।
अरुंधति, हाँ। पहले ये देखो ये है वही या नहीं है। उन्होंने कॉल किया था क्या आपको या
नहीं मेरे
किसी को?
पास कोई कॉल नहीं आया। शर्मा तो नहीं है मैम, खाली अरुंधति है।
एक मिनट रुक जाइए, वे वही है ना, बाहर के स्टूडेंट्स रहते हैं। माइक ऑफ है ना?
माइक ऑफ है।
मैं मैम को कॉल कर देती हूँ।
मैम ये सारे आपके डिपार्टमेंट के हैं जो आ रहे हैं जिनका रोल नंबर नहीं है?
वही तो वही चीज़। हाँ, एक कुछ एमडी स्टूडेंट्स भी होते हैं, दीपिका कुमारी, हाँ ये स्टूडेंट
ये
है।
अंजलि सिंह हो गया
ये
और
हमारे स्टूडेंट्स हैं।
यथार्थ मिश्रा हो गया, लीला लक्ष्मी,
हाँ, लीला लक्ष्मी वो एमबीबीएस बैच है वो।
सोनम सिंह जो है।
वो ये भी हमारे डिपार्टमेंट से हैं।
हम्म। तो जो बाहर करेगा तो वो रोल नंबर तो नहीं
वही
डालेगा
ना,
अपना?
वही वो रोल नंबर भी डाल रहा है अपना,
अच्छा।
नेम भी स्टूडेंट का डाल रहा है, तो हमें वो जानना है कि कौन सही है कौन गलत है, वो तो हम नहीं पता कर सकते ना। दिक्कत ये है। और कोई अरुंधति है क्या वहाँ पे? सिर्फ एक है।
अरुंधति?
हाँ, एक बार चेक करो आप। फिर मैं कॉल करती हूँ अरुंधति मैम को।
मैम यही है और नहीं है कोई?
गुड आफ्टरनून मैम, मैं डॉक्टर मोनिका बोल रही हूँ आपका डिपार्टमेंट। या मैम, नाउ इज योर क्लास ना फ्रॉम 2 to 3? हाँ, yeah, because there was a lot of problem yesterday. Some people were joining with two numbers IDs, so we were just confirming that it is you ना Dr. Arundhati, which it is shown over here. So that हाँ हाँ कर दो भैया, हमको मैम की प्रेजेंटर बना दो। हाँ ओके ओके, we're just making you the presenter. Okay, thank you मैम, बाय।
ठीक है मैम,
हाँ बना दो उनको।
चेक कर लेंगे मैम एक बार ना। हाँ भाई। हाँ नहीं, शर्मा जी नहीं है, खाली अरुंधति नाम से आया, डॉक्टर अरुंधति ऐसा करके आ रहा है। ठीक ठीक है। और सुनो, ये लॉक कहाँ से करना है ये?
डॉक्टर अरुंधति शर्मा को मनोज जी जरा प्रेजेंटर बना दीजिए।
बना दिया मैम। मैं
उनका लेक्चर 3:15 तक जब खत्म होगा ना, उसके बाद नेक्स्ट डॉक्टर स्नेहा को LT2 में आके करेगी।
LT2? अच्छा ठीक है मैम। डॉक्टर स्नेहा?
डॉक्टर स्नेहा गुप्ता वो LT2 में आके डिलीवर करेगी।
Ritu ma'am, hello.
Yes.
I'm Dr. Monica ma'am. I'm in the LT. So we have made the Dr.
Yeah.
Arundhati the presenter.
So she will start.
Yeah ma'am. Yeah.
We have to wait to join. There is one issue. The student had contacted me.
Yes ma'am.
His name is just a minute. His name is Animish Giri. 3641.
364. Yeah ma'am. Two people have joined with the same ID. I saw it in the morning.
Yeah, but he's saying that somebody posted the comment.
Yes ma'am.
On his behalf.
Yeah ma'am.
And he has been right now, Animish, you are there. Please unmute your mic and speak up.
Good morning, ma'am.
Good afternoon.
You are MBBS AIIMS.
Good afternoon, ma'am.
Are you MBBS AIIMS first?
Yes, ma'am. I am.
You are the
Yes, ma'am.
Okay. So can you give me
Yes, ma'am.
a message right away? Just send me a message. I just want to confirm your identity on my number. Just send me a message or call me.
Ma'am, on the GoToMeeting or on the WhatsApp?
On the WhatsApp. I want to confirm your identity.
Okay. Okay, ma'am.
Alright, fine. So now, Dr. Monica, if there is a second Animish who joins,
Okay.
just immediately log on. Okay?
Okay, ma'am.
Because apparently someone posted a message in the student's name and he was debarred by mistake.
Yeah, ma'am. I know 3 students of them have been disbarred.
Yeah. So now let's hope the episode which has been happening since last couple of days is not repeated. Let's
Okay.
hope for the best. Dr. Anviti, are you there?
I am there.
This was regarding some offensive music and offensive messages that have been repeatedly being posted yesterday in my class in the afternoon as well as during Dr. Nag's class in the morning. So some action. So just ignore them if they are there. Dr. Monica is in the LT. She will coordinate regarding blocking those miscreants out.
Yeah, sure.
Please start.
Yeah, please make me the presenter. Monica,
Yes, ma'am.
You but doesn't show up.
it is not showing on my screen.
हाँ, चला के दिया है।
Yeah.
क्या शो नहीं करता था?
नहीं, उनको शो नहीं कर रहा है।
नहीं, एक मिनट में करता हूँ।
हाँ, कर।
डॉक्टर अरुंधति। हाँ।
मैम एक बारी चेक करो एक बारी। अब कर रहा है मैम?
नहीं, अभी भी नहीं कर रहा है।
एक मिनट रुकिए, मैं वापस से करता हूँ।
डॉक्टर अरुंधति, हाँ।
No, it is not showing yet.
Yeah.
अभी उनको प्रेजेंटर नहीं बनाया गया।
मैम बना दिया है लेकिन वहाँ शो नहीं कर रहा, पता नहीं क्यों। मैं अभी चेक कर रहा हूँ।
आप अपनी स्क्रीन शेयर नहीं कर पाएंगे ना जब तक वो शो नहीं करेगा।
नहीं मैम वो कर रहेगा, एक मिनट रुकिए।
Dr. Arundhati, are you able to unlock your screen where it says my camera?
No, I can't.
अरुंधति कहाँ लिखा है?
एकदम टॉप पे चले जाओ पहले। एकदम टॉप
It
पे।
says ask the organizer to make you the presenter.
Right, that's right.
कहाँ देख देंगे जरा? अरुंधति
एकदम टॉप में जाओ ना पहले। आप
टॉप में गया था ना मैं अभी।
वहाँ से चेक करो पहले।
देखो ठीक है।
Because people are assuming fake identity even of the teachers. So that's why we have to be very careful and probably has to be able to change the platform or something. आपने लॉक तो नहीं कर दिया?
नहीं
वो राइट हैंड कॉर्नर से होता है ना, प्रेजेंटर बनता है।
नहीं वो मैंने कर दिया है मैम, ऑलरेडी मैम के सामने किया है मैंने अभी।
बट एक्चुअली मैम, हाँ
प्रेजेंटर का ना अलग से आना चाहिए ना, इसमें स्टूडेंटों में।
डॉक्टर अरुंधति वो आएंगी उनके।
the screen share option comes.
एक मिनट मैम कर रहा हूँ।
Ma'am, everything is switched on. Even the screen share is there, but Dr. Arundhati is not shown in the attendees list.
वही प्रॉब्लम।
No, in my she's there.
Here
In
it
my
is.
computer she is there, very close to where my name is.
एक बार नीचे जाओ।
Go down, please.
एक मिनट में पहुँचने वाले नीचे।
Beneath share और नीचे जाओ।
Shall I rejoin otherwise? Shall I leave and rejoin?
एक मिनट हाँ
इनको क्लिक करो।
ये ना
हाँ
It's my
राइट, हाँ
हाँ नहीं एक मिनट ये बनाता हूँ।
मेक द प्रेजेंटर।
ठीक है
यस कर दो।
मैम हो गया?
now she's there.
हाँ, अभी हुआ। हो गया।
अब होगा मैम, अब स्क्रीन शो कर रहा होगा।
ये
ठीक है मैम।
हाँ, अभी फिर गया ना?
चला गया?
दोबारा चला गया है।
फिर से गया अभी।
एक बार वापस से देखना
अभी
मैम।
दिख रहा है क्या मेरा स्क्रीन?
स्क्रीन ऑन करो।
हाँ वही कर रहा हूँ मैम, अभी देख रहा है?
नहीं, अभी भी नहीं दिख अभी 1 मिनट के लिए आया था फिर चला गया।
अच्छा
हाँ, 1 मिनट के लिए वो स्क्रीन का सिंबल आता है ना, to the presenter.
It had come but it has gone now.
That's gone immediately. Yes, that happened. आप फिर से प्रेजेंटर बनाइए मैम को।
1 मिनट में मैम आ गया।
ऐड करो पहले।
नहीं आया, नहीं आया।
अब आया।
हाँ अभी आया। ओके, आई एम क्लिकिंग ऑन दिस। या
1 मिनट, यहाँ ऑफ दिखा
हाँ,
रहा
ठीक
है,
है अभी।
स्क्रीन ऑफ दिखा रहा है।
और इस तरह
क्यों ऐसे हो रहा है?
everyone is joined. Dr. Anandati, please start. Thank you.
Yeah, I'll start. Yeah. Yeah.
एक मिनट चैट बॉक्स को
Right. Okay, so
बंद मत करो।
everybody's joined now. Let's start the lecture then.
इस पे
Okay. So yesterday what we did was, so good afternoon to all of you. So yesterday what we did was the
ये
Mendelian,
भी बंद कर दो।
yeah, we've done the non-Mendelian, we did the Mendelian pattern of inheritance yesterday. So today we'll look at some of the non-Mendelian patterns. So single gene inheritance was what we had seen, but it has been observed that other than single gene inheritance, there are some patterns which do not conform to the Mendelian patterns, that there is a divergence from them, and these are known as the non-Mendelian patterns of inheritance. So examples will be mitochondrial inheritance and some conditions like genomic imprinting and uniparental disomy. So we'll deal with them in detail. So first we come to the mitochondrial inheritance. So as you all know, we have within the cell, we have a nucleus, and the nucleus contains the DNA. So apart from that, we have cell organelles like mitochondria, which also have their own DNA. So now each mitochondria can have about 800 to 1000 or more than that DNA molecules. So the DNA which is contained in the mitochondria is circular DNA, and it has about 16 kilobases. And like this, thousands of circular DNA molecules are present within the cell. So if you look at the mitochondria, it is quite an independent body. It has its own DNA, and its own DNA can be replicated, which replicates by simple fission, like how the bacterial genome replicates. And it has about 37 genes. Out of these genes, most of the genes like code for whatever are the requirements of the mitochondria. That is because it is dealing with oxidative phosphorylation. Especially we call it as the ATP, or the powerhouse of the cell. So it is making enzymes which are required for oxidative phosphorylation. It has 2 genes which are coding for rRNA, and the remaining genes code for tRNA molecules. Now, the DNA of the mitochondria can also carry mutations, and those mutations will be similar to what we have seen in the nuclear DNA, like point mutations, deletions, insertions. Only thing that we have to remember is the mutation rate is very, very high in the mitochondrial DNA. That is because the mitochondrial DNA does not contain the proofreading. If you remember your basic biology, when DNA replication occurs, the DNA polymerase replicates the DNA. It also has a proofreading mechanism where it goes back and sees that whatever nucleotides have been incorporated, they are all the right ones. If it's a wrong nucleotide, then that is removed and the correct nucleotide is inserted back. So this is known as proofreading. So this proofreading is absent in the mitochondrial genome. So because of that, several mutations get accumulated in the mitochondrial genome.
यहाँ पे दिखा रहा है ना
So now when the mitochondria, when the cell is dividing, like the cytoplasm when it divides, even the mitochondria also divide. So its DNA molecule undergoes replication by simple fission, and all the mitochondria are distributed to the daughter cells by random distribution. There is no choice. Any mitochondria can go to any of the cells. Now suppose there are mutations accumulated in the mitochondria. A cell can get only mitochondria which are all normal. So if it is only of one type of mitochondria, we call it as homoplasmy. It can also get mitochondria which are carrying only the mutant genome. So that is also a condition of homoplasmy. But most of the cases it is heteroplasmy where there is a mixture of both the mutant mitochondria and the normal mitochondria. So a disease which is expressed as a result of mitochondrial mutation can depend, the severity of that disease can depend upon how many of the mutant mitochondria are present and how many of the normal mitochondria are present. So some of the diseases or disorders which have been documented as a result of mitochondrial mutations are what we call it as Lyon or Leber's hereditary optic neuropathy. Then there's a kind of epilepsy which we call it as the myoclonic epilepsy of the red rat fibers, Pearson syndrome, Leigh disease. So this disease, we have to keep it in mind that most of these mutations are affecting such of the tissues or organs which have a high requirement, high energy requirement. Okay, so like the heart, the eyes, etc. So this is very simple mitochondrial inheritance, and if we want to analyze mitochondrial genetics, it becomes very difficult because we do not know every cell is having so many mitochondria, which mitochondria are mutant, which are not mutant, we don't know. So the analysis of the disorder or analysis, if you want to say how many of the individuals might be getting the disease, it is very difficult to say. So counseling becomes very difficult. And I hope you remember that mitochondria are all, we inherit all the mitochondria from our mothers. So there is absolutely no mitochondria which is inherited from the paternal side. So the disorders I'm telling is these hereditary Leber's hereditary optic neuropathy. These are the examples. These are the disorders I'm talking about. Okay, somebody is asking me a question. So these are the diseases which are, nowadays there are many more disorders. One kind of infertility is also there which occurs as a result of mitochondrial mutations. So there are many. And another thing which we have to keep it in mind is as the human being is aging and the cells are dividing, due to age also many mitochondrial mutations get accumulated, and some of the diseases can be manifested at a later age. So in this, what you just have to keep it in mind is that there is a mitochondrial inheritance also, due to which some kind of diseases or disorders can arise. Now the second deviation is the X chromosome inactivation. So yesterday we just had an introduction to this. So if we discussed about Mary Lyon's hypothesis, where she had said that because females contain 2 X chromosomes and males have only 1 X chromosome, the dosage between the 2 sexes has to be maintained. That is why one of the X chromosomes in the female is inactivated, and effectively there is whatever genes are present on 1 X chromosome are only expressed at a time. So 1 X from the female and 1 X of the male. So there is dosage compensation. All right. So now how did people, or how did Mary Lyon or others understand that there is inactivation of one of the X chromosomes? So there have been several studies, like there is the G6PD gene is is present on the human X chromosome. So people have studied its expression. Suppose in the homozygous condition, both the alleles are expressed, what happens? If one allele is expressed, what happens? So that is how they have come to the conclusion that it is both the, if both the alleles in the females are never expressed, it is only one allele which is expressing. So another example is the calico cats, if you have heard about them. So this is a cat which has a patchy kind of body appearance with three colors: orange, black and brown or white in color. So what happens in this is there is a gene which is coding for orange and one for black. So depending on the X chromosome, so depending upon which allele is or which of the X chromosome is inactivated, that allele will not be expressed. So now here one thing you should keep in mind is every cell has two X chromosomes. Now every cell can make an independent choice whether it wants to inactivate one X chromosome which is coming from the maternal side or the X chromosome which it has got from the paternal side. So once a cell decides to inactivate one X chromosome, its successive daughter cells will always inactivate that particular cell. So if you consider a tissue, then it will be a mixture. Some cells are inactivating the maternal X chromosome, some cells may be inactivating the paternal X chromosome. That is why you get a patchy kind of appearance like this. And again, another example that I told you yesterday was sometimes the X-linked recessive inheritance, the females also express mild symptoms of the disease. This also happens because of the X chromosome inactivation. Suppose some cells have inactivated the normal X chromosome. So then what happens? The expression of the X chromosome which is carrying the mutant allele will be seen. So sometimes we have seen females with Duchenne muscular dystrophy who show signs of muscle weakness. So it becomes very difficult for them to sit down or get up. So this kind of patchy appearance is also seen among the human different kinds of disorders. Somebody is asking me about the Klinefelter. So I think you should just concentrate on what is being said. You will be dealing or you will be taught about Klinefelter syndrome and other chromosomal disorders also in the next class. So first understand this, so automatically you will understand what happens in Klinefelter syndrome. But just to let you know, like if there are suppose 3 X chromosomes, like Klinefelter has 2 X chromosome and 1 Y. So 1 X chromosome will be inactivated even in the Klinefelter syndrome. Alright.
So
Now
why
the
would that be of an issue? Like ma'am, I was asking that this inactivated X chromosome is expressed slightly or is it totally inactivated?
It is totally
Ma'am, or
inactivated.
is it like in case of Turner syndrome with that one X chromosome itself be heterochromatic? Is it something like that?
No, it is not like that. In the XC, whatever, you have variants of Klinefelter syndrome also. Some Klinefelter syndrome individuals can have triple XY. Okay, some can have 4 times X and Y. So one X is always active, kept in the active form, and the other X chromosomes are always inactivated. Okay.
Ma'am,
So
okay,
this
and I think we gave a special name, something as bar bodies or something, if I'm not wrong. So that is this condensed heterochromatin, right? Bar body or
Yes,
something,
yes.
I don't.
No, you are right. It is nowadays what happens is bar body is something which when we were not doing karyotyping, so that is how bar body was identified. So people used to say one of the X chromosome is heterochromatinized and it takes up a dark stain and that was known as the bar body. So one X inactivated chromosome is actually a bar body, right? So if there is Klinefelter syndrome with XXY, there will be 1 bar body. If there is triple XY, then there will be 2 bar bodies, like that. But what happens is, more than bar body staining, it is nowadays it's always better to do karyotyping where you see the number of chromosomes clearly. Okay, but inactivated X chromosome you will always see as a darkly stained heterochromatinized bar body. Okay?
Okay, ma'am. Thank you.
Yeah. Okay. So now we come to the second, that is genomic imprinting. Now, genomic imprinting is, if you people have seen, you must have posted letters somewhere. So what we do is we take an envelope and we paste some stamps, and you go to the post office. So what does the postman do? He will put his own stamp on the stamps that you have pasted, the 5 rupee or the 10 rupee stamp, isn't it? So what does it mean? So once he has pasted those, he has put his stamp, then nobody can reuse the stamps that you have pasted. So in a way, he is imprinting, he is stamping your letter so that nobody can remove your stamps and reuse them again. So similarly, in the case of our chromosomes or genes also, a stamp is put. Like, if suppose there is a certain gene, a stamp is put on that gene or a group of gene, so that those particular genes will be expressed if they are inherited from the mother's side, or some genes will be expressed if they are inherited from the father's side. So this is what we call it as imprinting. So imprinting means it is asking the genes, or it is putting a mark on the genes, whether they have to be active or whether they have to be silent. Alright, so this picture here is showing you the same thing, imprinting. And what happens is we have identified several genes, especially which are grouped on chromosome numbers 11 and 15, which are genes for imprinting. That means there are about 20 genes which have a role to play in imprinting, and these are very short genes, and these are very small genes, and they have very few introns. And it is said that these genes, which are present on chromosomes 11 and 15, they make their product, the RNA, mRNA, and this mRNA, they say, is not transcribed into any protein, but that will go and cover some of the gene or areas which have to be silenced. So those genes will not be transcribed, and their protein will not be made. So what is then genetic imprinting? It is, again, if you want to see, genetic imprinting is certain areas or certain genes, you just put an imprint, or you put a stamp, or you mark them, or you order them that you will not be expressing yourself, or you will be expressing yourself. So how is that mark put? That is put by the attachment of methyl groups. So if you have, you remember your methyl structure CH3, so methyl group is a bulky group. Now suppose this methyl group is attached to the 5' end just before your promoter region. So what of a gene? So what happens? It will occupy a lot of space, and it will not allow the transcription factors to come and bind, and also the polymerase to come and bind. So that particular gene will not be transcribed. So if it is not transcribed, we call it that the gene has been silenced. So it will not be able to make its product. Okay, so when does imprinting occur then? Normally it is said that imprinting is erased during early embryo development. So after fertilization, during the first about 4 to 8 cell stage, all imprints which were there in the DNA of the sperm and the ovum are all removed. Then as the embryo develops, and then the cells that start differentiating, and the cells which are destined to form the gonads, the cells which are going to form the sperm or the eggs, during gametogenesis, all the imprints are again put back. That is what we know as of today. So then you might ask, why should genes be imprinted? So gene imprinting is nothing but a regulatory mechanism. So now, if you remember yesterday, I told you an example, I gave you the example of Huntington's disease. So Huntington's disease always occurs at an age of 40 to 50 years. But the genes are there, the mutant genes are already there from birth. So why doesn't that happen? Why doesn't the gene express itself, or why don't the disease symptoms come at an earlier age? So it is because of imprinting, because one has to understand that there are, all the genes are there, but all the genes in a cell are not always expressing at the same time. Some genes are silenced and some genes are made to express. So how are those genes silenced? How do they remain silenced? It is because of the imprinting. And how is imprinting occurring? It is due to the addition of the methyl groups. So all the genes in the cell then are expressing themselves in a tissue and a time specific manner. So that is the reason why imprinting occurs. So that is a form of regulation to the expression of the gene. So this will study with respect to two typical examples of imprinting. So one is a Prader-Willi syndrome. So this Prader-Willi syndrome, individuals will be very short and obese with small hands and feet, mental retardation, etc. So in this case, what happens is, if the chromosome number 15, so there is a small deletion, a small region of chromosome 15 is deleted in the father. Okay. Now, the same thing, what happens, the mother's chromosome 15, this particular region is imprinted. So all of us actually have an imprint in the maternal chromosome or the paternal chromosome, and they will either express themselves or not express themselves. So when everything is normal, we will never come to know about the imprints. But there was a condition of Prader-Willi syndrome where they found that the father carried a mutation in this region and the mother's same region was imprinted. So practically what happened, whatever genes were present in this region on the chromosome, there was total loss of information in this individual. So because of this, it results in a condition called as the Prader-Willi syndrome. Now just the opposite of this, we have another syndrome known as the Angelman syndrome. So this is unlike the obese personality. Here we have a hyperactive personality with this is also known as a happy puppet syndrome. So puppet-like movements and absence of speech, excessive laughter, etc. Now in this case, what happens? Just the opposite. The maternal region of chromosome 15, this is deleted, and the father's 15 chromosome is normal, but it is imprinted. So because it is imprinted, this individual then will not have information again for that same region. So it will result in a condition called Angelman syndrome. So once this condition was discovered, then it was like people understood, the scientists or the researchers understood the importance of the paternal origin of the genetic material. So then you will ask, why should it have two different kinds of conditions? Why should it express as Prader-Willi and as Angelman syndrome? So when people try to isolate DNA from this particular region, 15 chromosome band 1 to band 3, it is a long stretch of DNA, and then they saw that it contains several thousand genes. Now what happens normally in all of us is, there are certain genes which are expressed only from the mother's chromosome 15 and certain genes which are expressed only from the father's chromosome 15. So in these two conditions, what happens if there is a deletion? There is a total absence of information. That is why it results in two different conditions, two different syndromes. Is this clear to all of you? Yes. Yes, but this is an autosomal disorder. This is not a sex-linked disorder. It is 15 autosomal. All right. Okay. So then we move on to another deviation, that is mosaicism. So mosaic, you all must be knowing, we have mosaic tiles, that is existence of 2-3 different types in a if a mosaic tile, if you have a tile, then you have 2-3 different colored inclusions in the tile. So in this, if you see the tissue, you have 2 different kinds of cells. Now in once, I'm just showing you this as an example. This can be a mosaic where in the tissue, one of the X chromosome, like the maternal, is inactivated, or in the other, in some cells, the paternal X is inactivated. So this we can call it as a mosaic pattern. But suppose when you later on, when you do the chromosomal disorders, you will come to, you will see many conditions where mosaicism exists in our body. Like what happens, there can be some cells, especially in the case of Turner syndrome or Klinefelter syndrome, where you have some of the cells carrying the normal, like 46XX normal condition, but some cells can be carrying just 45X, only one X chromosome. So mosaicism can occur in the body. Just opposite to mosaicism is, like, if you want to define mosaicism, then it will be existence of 2 to 3 different cell lines in an individual, but they are derived from the same zygote. Like it is the zygote which is after fertilization, the embryo that is forming, and some of the cells which are dividing. Sometimes one of the cells, they can non-disjunction can occur, and one of the cells can get 1 chromosome less, the other cell can get 1 chromosome more. So the descendants or the daughter cells will again have the same 1 chromosome less or 1 chromosome more. So like that, there can be a mosaic pattern. Now mosaicism can be somatic mosaicism, that is, it can develop at a later stage in some of the cells. So it will not affect, it will, the individual who is carrying the somatic mosaicism, only he will be having, or he might show a segmented or a patchy kind of appearance with respect to some cells or some chromosomes. But okay, to give you examples, again, neurofibromatosis, if you remember, the disease of the nerve endings, which showed variable expression, it can be mild, moderate, or severe. So sometimes some individuals can show just mild symptoms in one part of the body, moderate or small neurofibromas in some part of the body. So depending upon the cells that are carrying the mutations, the disease can be manifested as mild, moderate, or severe. Now, so mosaicism can be, again, as I said, somatic, and the other one can be germline mosaicism. So germline mosaicism, to explain to you this particular phenomenon, I have an interesting story to tell you. So what happened was, this particular person, a man, he was married and he had a child, a son. Now this particular son developed this osteogenesis imperfecta, what we called, we studied yesterday. It's an autosomal dominant condition, okay, with a mutation in the collagen gene. So now what happened was, this person had a mutation. So when they took him to the clinician, he was asked to undergo a diagnostic test to confirm. So this person had a mutation, the son. The parents were tested. Parents did not carry the mutation. So there was nothing they could do, so they went back. So now what happened, after a few years, this person came back again. He had in the meanwhile divorced his first wife and had got remarried, and he had a daughter from the second wife. And it so happened that this daughter also developed osteogenesis imperfecta. So he again went to the clinician. So then that was the time when they said, when they tested this lady also, she was also negative for the mutation, he was also negative. That was the time when this phenomenon of germline mosaicism was discovered. So what they did was they took a gonadal biopsy, a small tissue from his gonads, and that they extracted the DNA and they tested for the presence of mutation in his gonadal tissue. And it was found that this particular person, the father, was indeed having germline mosaicism. Like some of his cells were carrying the mutation in the collagen gene, and while the remaining cells were normal. So that is why it resulted in his children being born with the disease, that is osteogenesis imperfecta. Now it was a chance that both the children got his disease gene. He could have a normal child also. Okay, so it so happened that both the times the sperm that fertilized the ovum was carrying the mutation. He could have had a normal child also. So germline mosaicism will be then. How does it occur then? So the cells which are destined to form the gonadal tissue, if in one of the cells a mutation occurs by chance during replication of the DNA, then that particular mutation will be restricted to the daughter cells of that particular cell. So that is why finally the gonads that are being formed, so all of them are not carrying all the cells are not carrying the mutation, but only a few cells are carrying the mutation. So that is germline mosaicism. Is this part clear? Yes, yeah. Okay. Okay, so the fourth deviation will be uniparental disomy. So disomy is a condition where we have two chromosomes. Uniparental means you getting both the chromosomes from one parent only. So when can you think uniparental disomy can arise? It can arise when parents are unaffected, but the child gets a recessive disorder. When will the child get a recessive disorder? Only when the parents are carriers, right? So parents are carriers, both the parents should be carriers. But suppose there is only one parent who is a carrier and the other parent is normal and the child gets a recessive disorder, then one can think that it could be uniparental disomy, because both the chromosomes carrying the mutation the child could have received from the parent, from the single parent. Alright, so same thing, and there are two types of uniparental disomy, isodisomy and heterodisomy. This I'll explain with the help of this diagram. Okay, so this is isodisomy, that means both the chromosomes are being inherited from the single parent. Now if you look at this is one parent carrying two chromosomes, the other parent. So during reduction division, first meiosis, each cell will get one parent, the one chromosome. Now during meiosis 2, what can happen? These will divide into the chromatids and each chromatid is received by each cell. Now due to non-disjunction, if one of the cells doesn't receive one chromatid, both come to the other cell only, then this particular cell is having effectively one whole chromosome from one parent. Okay, now incidentally, if this is carrying a mutation over here, the mutation will be there occurring double dosage in this particular cell. Now if this gamete is fertilized by a normal gamete, then what happens? You have a trisomy condition for that particular chromosome. So here natural selection will act and it will try to remove one of the chromosomes. Now natural selection just knows that there are 3 chromosomes, so it has to act. It doesn't know which chromosome has to be removed. So suppose it removes one of these chromosomes, then the normal condition will be restored. One of this chromosome, one of this chromosome. But suppose it removes this normal chromosome, then again these 2 chromosomes carrying the mutation will be remaining in the embryo. So then there will be a double dosage which is resembling a recessive condition. So this individual might then show the disease. Okay? Is this clear? Alright. So heterodisomy heterodisomy then will be a condition where the non-disjunction can occur at the stage of meiosis 1. So now the first reduction division, when the chromosomes are being separated into the daughter cells, due to non-disjunction again, one of the cells may not receive one chromosome and both the chromosomes are received by the other cell. And similarly, here the normal thing, these 10 cells will die off, and this particular, again there is a normal disjunction. Okay, so again a condition of trisomy will occur for a very short period of time. Again, one of the chromosomes has to be removed or weeded off. So depending upon which chromosome is removed, then it will be whether it is the fetus form will be normal or it will be abnormal. So uniparental disomy is generally seen in many conditions like Duchenne's muscular dystrophy or even Prader-Willi syndrome, etc. So these are all very rare events, but they have been documented. All right. So when does loss of chromosome occurs just like that. The chromosome is removed from the cell. Okay, it is okay. So when do you consider your uniparental disomy must have occurred? That is when a rare homozygous recessive condition occurs with only one heterozygous parent. Like the child has a recessive condition. You test both the parents. One parent is normal without any mutation. He is not a carrier. The other parent is showing the mutation. He is a carrier. Okay. So with one parent, you should not have a recessive condition. So in such cases, then you can think that uniparental disomy must have occurred. Or if some of the rare recessive condition disorders you see with unexpected features like mental or growth retardation. So some, like a rare recessive disorder, like suppose cystic fibrosis is very common, like you say you take alkaptonuria or some metabolic disorder. If you see, it should not be associated with say growth retardation or mental retardation. So if you see something like this, some unusual features, then you can think uniparental disomy must have occurred. Or suppose in an X-linked recessive condition, father to son transmission is seen. Okay, then also you can think that it could be a rare, it could be uniparental disomy. All right. Is this part clear? Then we move on to the next portion. So if this is clear, we'll go ahead. All right. So now we come, what we have done is Mendelian inheritance or single gene disorders, then non-Mendelian. Yes?
Could you explain the last point from the previous slide?
Last point. Okay. Females with normal fathers affected with rare X-linked recessive disorder. Now female will always get the X chromosome from the father, one X chromosome from the father and one X chromosome from the mother. Okay, so now suppose a female is showing an X-linked recessive condition but has a normal father. But then what happens? Then she must have got, her mother might be a carrier, and she must have got the two carrier chromosomes from her mother only. So then it becomes recessive condition in her case, homozygous condition in her case, and she will express the disease. Is it clear?
From the carrier mother, she will get isotype of inheritance.
Yeah. Yeah, from the carrier mother, like carrier mother, from the carrier mother, we do not know her mother is a carrier, no, because she is not expressing the condition, X-linked recessive condition. Father is normal, but if the child gets the X-linked condition, and the female gets the X-linked condition, then immediately you will think that it has to be in double dosage, but the father is normal. So then in such cases, you have to consider that the mother may be a carrier, and mother, both the mutation carrying X chromosomes she has received from her mother. That is why she is expressing the disease. Okay?
Excuse me, ma'am, I have one doubt in this part only.
Yeah.
Single point being that you mentioned that it has to be a rare disorder. Why especially rare only? Why not the common ones, other common genetic disorders?
Because these events which are occurring are very rare events. A uniparental disomy also, it is occurring in a very, very rare condition. Okay, so that is why we are saying rare disorders. Okay, like normally there are so many recessive conditions, so we do not see uniparental disomy occurring in those conditions. Okay, so that is why we are saying rare, because some, as compared to the dominant disorders, they are very rare. But recessive disorders, as compared to dominant disorders, will be comparatively common. Okay, but overall, single gene disorders are very rare. Getting the point?
Yes, ma'am.
Yeah. Okay. So now we have seen about single gene disorders, and so I think in your all these years of your life, has any one of you come across any patient with Huntington's disease or cystic fibrosis or X-linked mental retardation, any of the single gene disorders? Suppose you have gone to the hospital or somewhere, have you seen anybody? I don't think any one of you has seen, isn't it? But I'll ask you another question. Now, how many of you have seen, so how many of you have seen patients with diabetes and heart problems and high blood pressure and obesity? How many of you have seen? All of you have seen, isn't it? So that is the difference between genetic disorders or Mendelian disorders and complex disorders or multifactorial disorders. Okay, so if you go to any hospital or if you go to any, you will find most of the people who are affected with these conditions only. So that is why these are occurring very, very in a common manner. And if you remember the spectrum that I showed you yesterday, one end the genetic diseases, the other end environmental diseases, and in between was the multifactorial diseases. So that is diabetes, etc., high blood pressure, etc. Okay, so these are the conditions which are governed both by genetics as well as environmental changes. Now, if you can see this, you must have studied about polygene or polygenic inheritance in the lower organisms. So in human beings, we normally don't call it polygenic, we call it multifactorial inheritance only. So what is polygenes then? If a single character is being governed by 2 or 3 different genes. Like I'm giving you an example of skin color here. So now skin color is governed by 3 genes. 3 genes, each gene will have 2 alleles, isn't it? So now depending upon what alleles a particular person is having at the 3 genetic loci, his skin color will be governed. So now in a population of like suppose you take Delhi population, if we are able to type the genes or look at the alleles of each and every person, and if we try to plot that, then we will get a curve like this. Do you know what this curve is known as in statistical terms?
Bell-shaped.
Yeah, bell-shaped curve or a Gaussian distribution in a population. Okay, so in polygenic, then what happens is there will be small, each gene will be making its product, and that product, final that you see, the skin color, will be additive or cumulative effect of all the gene product. No one particular gene is not dominant or not recessive to the other. So ultimately what you see is each gene is contributing, and then what you see, the final result will be the additive effect of all the genes. Similarly, here I am showing you eye color also. See, eye color only 2 genes and 2 alleles. Okay, so but you see the difference, like right from dark brown color to light blue color. So this is how because of the additive effects of the alleles of the genes, you can get different eye colors. That is why this is also known as quantitative inheritance. Okay, so like if you see height of an individual, height in a population, or intelligence in human beings. Okay, so these are all, if we try to plot them, I'm showing you here what we call it as a continuous phenotype, because you are not going to get height of individuals in discrete variables. We will not have a person with 5 feet of 5 feet 1 inch, 5 feet 2 inch, 5 feet 3 inch, like that. You will have a continuous variable, isn't it? Even blood pressure. So these are all what we call it as bell-shaped curves or Gaussian distribution or a normal distribution in a population. So what we are studying under this is under multifactorial inheritance. Now, if you remember our first class where I said we can study genetics at different levels. So till yesterday we studied at the pedigree level, at the family level. So what we are doing now is at the population level. The population can be a small population of your first year MBBS batch, this year's batch. It can be a small population of the Department of Anatomy, or it could be a population of the whole of AIIMS. So whatever population we are studying, if we measure any particular characteristic and we try to plot it, it will always try to assume a normal distribution. Is this point clear? Okay, so how do you then these, if you want to look at the traits, then 1 minute. I'll come to this slide first. So suppose now, because we do not have, alright. So now suppose we want to study a qualitative trait or a quantitative trait. What happens is, in a multifactorial condition, it will be governed to certain extent by genes and to certain extent by various environmental factors. Now those environmental factors, we do not mean the weather conditions. Okay, by environmental factors we mean everything other than genetics. The food that we consume, our lifestyles, our different small and big stressors that we have, the micro environment where we are staying, the macro environment where we are staying, everything contributes to the environmental factors. So now if you want to study multifactorial inheritance, then it becomes a little difficult, because how are you going to study? Isn't it? So what people have done till now is they have tried to study in terms of familial aggregation. So what, or they have used case control studies, or they have used twin studies. So what does it mean? So familial aggregation means, let's take the example of diabetes. Okay, so if you, each one of you try to remember or try to think of your own family, say diabetes or hypertension or heart problems, you try to think of your own family. You might be having 2 or 3 people in your family who are affected with that condition, isn't it? But now suppose you want to say that my father or my mother has diabetes, my grandmother also had, my uncle has. So suppose somebody asks you, what are your chances of getting the condition? So will you be able to tell them, or will somebody ask you, will you be able to tell what is your percentage of getting diabetes? You will not be able to tell. So because there are no ways by which we can assess, we do not know how many genes are there which are contributing, what are the environmental factors which are contributing to our getting the disease. So then people, when they have been studying all these conditions. So in the past a few years, like 50 to 60 years, most of the Western populations have a very good documentations of whoever has been born. So whether it was a single birth or a twin birth, so they have got very meticulously, they have actually registered their data. So people have done these twin studies, that means twins who were born. And suppose there is a history of diabetes in the family or hypertension in the family. So what they do is, if they are monozygotic twins and if they are dizygotic twins, so they will follow the twins right from the birth to the age of 50-60 years and see what are the conditions that the twins are developing, what are the diseases that they are developing. Okay, so like that one can do twin studies. One can do case control studies, that is, you study all individuals with, say, hypertension, and you study and compare them with all individuals who are normal, who do not have hypertension. So what do you want to study in these? You can choose whatever you want to study. You can look at some genes, you can look at the conditions in which they are staying, you can look at their food habits, their lifestyles, and try to do the comparison. Okay, so another study, okay, when we do the twin study, then you have two types of twins, monozygotic twins and dizygotic twins. So monozygotic twins are as good as being the same individual, and dizygotic twins will be as good as being two different individuals. So there is a term, we call it as concordance. Concordance means how much of similarity is there between the twins. So if they are monozygotic twins, then we think that they are sharing the same genetic makeup. So if one of the twins is having a disease, then the other twin also should have the same disease. Then we say that they are concordant twins. But you will not see much of concordance in the dizygotic twins, because they are as good as being two different individuals. Their genetic makeup is also different. Okay, so concordance is one thing which they say. If for a disease, like suppose if it is sickle cell anemia, if you're going to study in the monozygotic twins, then both of them will be 100% concordant, isn't it? But the dizygotic twin, one might receive the mutant allele, the other may not receive the mutant allele. So one can be normal, the other may not be, may not have the disease. Okay, so concordance is something which we say how. If for a particular disease, if the monozygotic twins are concordant, then it is what you are expecting. But suppose they are non-concordant, then you say that it is less of genetic factors and more of environmental factors. So other than case control studies and twin studies, another study which people do for this multifactorial conditions is adoption studies. So what we do in adoption studies is, suppose there are two families, say, one say in the, just for example I am telling you, somewhere in the north of India, say Kashmir, and there's another family, say in Kerala, and both of them, they exchange their children. So the Kashmiri child is adopted into a Kerala family, and the Keralite child is adopted into, say, the Kashmiri family. Now the Kashmiri family may be having a history of hypertension and heart problem, and say the Kerala family has a history of some other disease, diabetes or some other disease. Okay, so now the children at the early age, they are given off for adoption, Then say after 20, 30 years, 40 years, they will see who is developing what kind of a disease. So if the Kerala family child who are adopted into the Kerala family child is developing hypertension, then what do you understand from that? That particular child has more of genetic factors which is making him develop hypertension and heart problems. But suppose he doesn't develop that. So what does it mean? The environment is totally different. He might be having some genes, but those genes are not sufficient enough to cause the heart problem in the child. Is this part clear? The adoption studies. So people have separated in twins also at birth. Okay, so there was a study where they separated two identical twins at birth. One grew somewhere in the UK, United Kingdom, and one grew in the United States. So after 40 years, they studied the twins again, and they found that the twins had the same habits. Both of them liked coffee and strong coffee, and both of them had the same they like whatever likes and dislikes, they had the same preferences. Okay, and they studied for the diseases also, and they found that they were having the same diseases also, conditions. So this is how people have been trying to study and trying to, what do you say, delineate what are the genetic effects and what are the environmental effects for the causation of a particular multifactorial condition. Okay, so what I just told you in the form of adoption study is this, like all of us have certain genes. We have all genes which cumulatively may add up, and we might get diabetes or hypertension at a later age. So what does it mean? We do have the liability to get the disease, but all of us may not manifest that particular. No, environmental factors, and you will be studying this later on. There is something called as epigenetics. So some of the environmental factors can, what we just studied in the form of methylation. So some of these environmental factors can actually make a cell, a particular gene, to be active or to be silent. So environmental factors have that power. Okay, so this you will be doing this later on. So now here you understand that all of us have a liability to get a disease. So this is the population curve. So if you want to study, suppose a particular disease you are studying in the population, then you will have a small percentage, like you take our AIIMS population only, and if we try to see how many of them are having hypertension, you will see this, you may put the blood pressure values here and the number of people over here. Again you will get a curve like this, and only a small percentage of the people are actually having high blood pressure. Okay, so we call this as the population incidence, or AIIMS population incidence of people having hypertension. Okay, so what is happening? Everybody is having those small genetic factors, and everybody is also having the environmental factors, but all of them will not manifest the disease condition. All of them will not show hypertension. It is only those individuals in which the liability is more, or those individuals who have a problem, suppose they have more stress or more lifestyle conditions are different, those will be expressing the condition. So threshold traits means in a population, a certain point, now suppose there could be a pressure, high blood pressure here, say 160/90 or something. So beyond this, whoever is expressing, those people, this is known as a threshold of the population. So these are the people who are having high blood pressure in the AIIMS population. Alright, so you just understand that whatever you plot, whatever you measure and you plot for a population, it will always try to take a normal shape. And beyond that, if somebody is expressing the condition that you are studying, that point is known as the threshold point. And liability is all of us are having that particular requirement. All of us have those small genes, but they are not adding up to produce the. Now, even at this stage, the mean also, these individuals are having some of the factors which will make them get high blood pressure, but they are not adding up to manifest. Now, these individuals, suppose something happens to them, they have strong, problems, stress problems, so they can develop high blood pressure at a later point in time. Okay, so the same thing, threshold model of disease I'm showing you here. This is showing you the neural tube defect. So what it means is, any neural tube defects is multifactorial. One of the factors is lack of folic acid, also deficiency of folic acid. So when anybody can have a child which is being born can have the tendency or the liability to get a child with neural tube defect, but only when that threshold is passed or surpassed, then only that individual will be showing that particular condition. So if it is a Mendelian disorder, autosomal dominant condition, what will you say if suppose one child is born with the disease? Somebody will ask you, what is the chance that my next child will also have the same condition? So because if it's autosomal dominant, you will say it is 50% chance. Similarly, if it is autosomal recessive, you will say 25% risk. But for a multifactorial condition, you will not be able to give any risk, because how do you know what do you know, how many genes are there, what environmental factors are there? So we do not know what is the recurrence risk. So in such conditions, what we use is the empirical risk. So what are empirical risks? These are risk estimates which are derived by direct observation. That is, you take the population of Delhi. Now go and see how many children are being born with neural tube defects. Okay, so now in this particular year, so many children have been born. Last year, so many children have been born. So based on this, they do a statistical analysis, and then we are able to predict as to what is the chance that the subsequent child might be born again with a neural tube defect. So, but what you have to understand here is, these empirical risks will be very restricted to a particular population. Like Delhi population, the empirical risk for getting a disorder will be say X. But similarly, you go to Punjab or you go to Madras or Tamil Nadu, you will not, the same empirical risk will not hold true. Okay, so then the empirical risk will be, you have to observe that population over there, and then you have to derive the risk for that population. Okay, this figure is just showing you, this is the first curve here is showing you the population threshold that I told you, the liability. But if you, same thing, if you try to measure in a family, then you will have 4 or 5 people who are affected with high blood pressure. Then in them, the recurrence risk will be high, because they are all sharing the same micro and the macro environment in the family. So you will find more number of people affected with the condition. So for them, this threshold will be little high. Okay. Now, this is one term which I don't want you to go into the details. Just remember what is heritability. So there is a difference between inherited and heritability. Inherited means the mutations which is inherited from one generation to another. But heritability is just a measure. It is given by N square. It is just a measure which tells us how much is the contribution of genetics to a particular condition and how much is because of the environmental factors. So this is actually measuring the total genetic variance by the random environmental variance and the environment within the family and the total genetic variance. You need not go into the details. So X square, if it is between 0 and 1, more towards 1, you more than like 0.6, 0.7, 0.8, you say more of genetic factors are involved. If it is somewhere from 0.1 to 0.5, say, you can say that it is more of environmental factors which is causing the condition. Alright, so these are some of the various multifactorial inheritance diseases. Sorry, these ones. Multifactorial, as the name indicates, multiple factors. So multiple factors will be genetics also and environmental factors also. And by environment, we are encompassing many factors. Your food habits, your lifestyle, your stress points, everything. So multiple factors are actually contributing to a certain condition like diabetes or hypertension. Is it clear? Somebody asked what is a multifactorial? How to define a multifactorial condition? So multifactorial means multiple factors. Okay. So now this part is important that you have to remember for a multifactorial. Yes, if you remember the last slide of our yesterday's class, I showed you 3 figures where I said everything is multifactorial, but in single gene disorders or Mendelian factors, then you have 99.9% or more, or majority of it is due to mutations in single genes. Okay, and there could be some other genes also which are involved. So the multifactorial or the environmental factors will be having a lesser effect in single gene disorders, but in complex condition or multifactorial conditions, the environmental factors will also be having a major role. Okay, so actually for us to understand, us means not only you and me, for the whole world to understand, we have divided this into Mendelian inheritance and complex inheritance. But environmental factors or multiple other factors also have a role to play in the inheritance of even the single gene disorders. That is why you find so many exceptions. Why did you see lack of penetrance? That is due to some other factors which is modifying those genes and not allowing them to express the disease condition. Is this clear? Whoever asked me the doubt? Okay. So now concentrate on this, the hallmarks of multifactorial inheritance. So now, if you remember, I just told you recurrence risk. So here, by recurrence risk, we have to depend upon empiric risk of that population. So now I am telling you the first hallmark or the first characteristic of multifactorial inheritance will be in a family if a child is born with one particular multifactorial condition. Here I am giving you a disease, ventricular septal defect, or it could be anything. Suppose there is a person who is born with a kind of multifactorial inheritance. Now, in the second pregnancy, the second child also being affected will be, say, 3%. If there are already two children born with the same kind of multifactorial disorder, then the third also being born will be 6%. So this 3 and 6 is just examples. It again depends upon the empiric risk of that particular population. So what you have to remember here is, if a family already has one child being born with, say, ventricular septal defect, then the chances of the second child also having the same defect will be increased. If the family has already two children born with the same defect, a third child also being born with the same defect will again increase. Okay, so higher the recurrence risk will be higher if more than one family member is affected. Okay, so now the second feature is the recurrence risk will be higher if expression of the disease is severe in the proband. So for this, proband means propositus or patient or index case. Okay, so if suppose in a family a child is being born with cleft lip, now the cleft lip severity can be like this or more deeper, and it can involve the cleft palate also and deeper also. So suppose a child is being born with a severe form of cleft lip and cleft palate, then to the same parents, the second child also being born with the same condition will be higher. Okay, are these 2 points clear?
Yes,
Type
ma'am.
in your okay.
Ma'am, in the diseases which depend only on genes, genetic diseases, each birth is taken separately. Means 50% chance of suppose if it is autosomal recessive disorder and both the parents are heterozygous, then there will be 25% chance, and that percentage remains same in case birth or fourth birth also. But in case of multifactorial inheritance, why does the percentage increase? And how does it initial births affect the next birth percentage of being affected?
Why it increases? In that case, what happens? We know there is only one single gene which is causing the condition, and if the gene is present in one copy, that is, and if it is dominant, it is going to cause a disease. If it's recessive and if the gene is present in two copies, it is going to cause a disease. But in these cases, multifactorially, we do not know how many genes are there which are having a change. We do not know what are those genes which of those genes are being manipulated by environmental factors. We do not have any idea about this. What we have idea is what we are able to see, what we are seeing in our population. So in the population, like suppose there is this Delhi population. So if we see an increase in the neural tube defects, okay, so then we see that those the mothers who are living in Delhi, they might be having some defect, or they are not having a defect. There could be some factors which are acting in such a way that there could be some foliage deficiency in the diet, say, or whatever food they are eating, maybe there is a deficiency in that, or you have so much of pollution over here, we are inhaling those chemicals. So these are all actually going to go and affect they might be affecting our transcription of the particular genes. So that is why if in a condition, like now in a family, all the families in Delhi will not have a child who is born with cleft lip or cleft palate. Only a particular family, say, has a child being born with that condition. Now, what does it mean if that condition, that child has, in that family, that child is born with that condition? It means whatever genetic factors and environmental factors which are required for the development of that clefting to occur are already present in that family. Isn't it? Family means the mother and the father. Okay? And if they are already present, then one child has been born with that particular condition. Okay? But in the subsequent condition, then what will happen? You know those factors are present. So it is all the more there are chances that that person, the second pregnancy also can result in a similar condition. But many times what happens, like we can talk in terms of neural tube defects. So one of the reasons I said is folate deficiency. So suppose the mother has one child being born with the condition. So immediately because of that, during her next pregnancy, she'll definitely visit the hospital. And once the doctors know that there is a chance of this occurrence in the family, at least they know that they should put the woman on folic acid supplementation. So at least one part of it we have taken care of. You're understanding my point? But suppose there are some unknown environmental factors, we won't have a control over them. Correct? Is it is it clear to you? Okay. Now, the third point is recurrence risk will be higher if the proband is less commonly affected sex. Now, this pyloric stenosis is a condition where there is an obstruction at this juncture, and it has been seen, and it is surgically corrected. A minor operation will clear this, so that the food from the stomach passes on into the small intestine. It has been seen that this pyloric pyloric stenosis occurs more in the boys, the male sex, as compared to the females. This has been data on observation. So why this is occurring, we do not know, but all over the world this occurs. Mostly the males are affected. Now, so what does it mean? Whatever is causing this pyloric stenosis to occur in that particular family member, so they might be having those particular genes and the environmental factors also present, that why a person like this is being, or a child is born with this kind of a problem. So suppose in this particular family, if now the female is born with pyloric stenosis, what does it mean? It means the rarer sex is born. That means all the necessary genetic factors are there in that particular family. So the risk in the subsequent pregnancy will be much higher. Okay, so it means that the males are having, if the male is being born with this, there are some factors which are present, which is making the male to be born with this condition. But for a female to be born with this condition, more higher number of factors should be available. Then only the female also will be born with this condition. So suppose in that family, a female is born with pyloric stenosis. That means whatever genes which are there acting for the formation of pyloric stenosis to occur, along with the environmental factors, are already present in the family. So the chances of the subsequent child also being born with this will be much more higher. Okay? But these are the three cases where the recurrence risk will be higher. But recurrence risk will decrease rapidly if the relatives are related, they are far or distantly related. That means in the nuclear family or joint family, you might find that there are more chances. But suppose at the second cousin level, third cousin level, fourth cousin level, then the risk will reduce drastically. So this is showing you again a neural tube form of neural tube defect. In the first degree relatives, the chance can be 4%. In the second degree second order cousins or second degree relatives, it will fall to 1%. Third degree and more, the chances of them developing this condition will be less than 0.5%. So what do you understand with this? It's the close family. They are sharing everything which is common. But as the relatedness decreases, like you go to second cousin levels, so they are not even staying in the same house. They may not even stay in the same city. So they are staying distantly. So their conditions, the environment conditions are also changing. their immediate microenvironment in the house is also changing, and the food habits also might change depending upon where they are staying. So that is why the recurrence, the risk will decrease rapidly as the family is being remotely or distantly related. But if you consider autosomal dominant condition or recessive condition, whether they are first cousins or whether they are fifth or sixth cousins also, if there is a mutation in the parent, then definitely the child is going to get. How much they are related to each other does not matter here at all. Is this clear? So now, the last point here is the prevalence of the disease. Suppose the prevalence of the disease is F in a population, then risk for the offspring will be subsequent children to be born or SIPs to be born will be square root of F. That means suppose the incidence of say a particular condition like say neural tube defects in Delhi is 1 in 1000. Then for a population, what will be the risk of a subsequent child being born will be square root of that. So that comes to about 1 in 32 or 3%. That is square root of 1 by 1000. So this is how normally you go on document the people are affected in a population, and one tries to derive at an empiric risk for a particular disease for a particular population. Now empiric risk will be different for different diseases also. It will not it does not mean that Delhi population, if 3% is the empiric risk for say neural tube defects, 3% will not be the empiric risk for say cleft lip or cleft palate. So it will differ with the disease and with the population of the area. So is this clear? We are finishing off now. We've come to the end of our topic. So if there are any small doubts, we can take up now.
लॉक करना है कहाँ ज्वाइन कर ही नहीं रहा वो अनलॉक कर दिया कॉल कर दिया उसको
देखना अलीशा कहाँ है
It is not 32%, it is 3%.
शो कर देगा वो
It's 3%, not 32%.
क्या नाम है
अलीशा
Yeah, no problem. Okay, so I think when you this you have heard for the first time, so I think whenever you get some time, if you just go through this topic, then you might understand better, or you might get some doubts also. So you feel free to communicate anytime. We have our group, the WhatsApp group. You can always ask your doubts on that. No, we are not following any population genetics book. The Bible for genetics will be Thompson and Thompson, and the other book on medical genetics by Jolly. Both books are equally good, but Thompson and Thompson more or less covers everything. So try to follow that, or Jolly, whichever you get this thing. Okay? Alright then, see you all. Have a good time and wish you all a very happy new year. Alright, so bye bye.
Thank you, ma'am.
Thank you to all of you.
Excuse me, ma'am.
Yeah?
Ma'am, where are you taking square?
That is how
Hello?
it is calculated. Yeah, that That's how it's calculated. That's a formula to calculate. Okay, root of f. If a prevalence of a disease is f, then root of f will be the what do you call the rate of prevalence at a particular time. Okay, that's a formula. All right.
Okay, ma'am.
Yeah. Okay, then I'm leaving the meeting, so your next class can continue.
नहीं बताया फेक है वो, एक मिनट अक्रम करके बताया था। एक मिनट में स्टार्ट हो जाएगा पर लेकिन गलत कमेंट्स वगैरह करेगा ना।
ट्रांसफर ऑलरेडी
एक मिनट,
कोई वेटिंग नहीं है ना?
नहीं अक्रम से कोई भी आया था मेरे पास मैसेज, अक्रम है वही। ओके
अभी पहले पीपीटी ओपन करो, ये बंद करो, कैमरा ओपन बंद करो,
कैमरा बंद,
पीपीटी ओपन करो,
स्क्रीन शेयर करनी पड़ेगी।
फर्स्ट ओपन योर पीपीटी।
चेक कर लो।
सुशील जी, आपको यही करना है कुछ
क्या?
इस क्लास के बीच।
मैं सुबह से बैठा हूँ जब से ही हूँ।
नहीं, ये नाम का पहला क्लास।
अच्छा ओके।
हाँ,
ठीक। ठीक।
आप यहाँ बैठ सकते हैं।
नहीं मैं बैठ जाऊँगा, ये चेक कर रहा हूँ ना अभी। ठीक चेक कर रहा हूँ।
हाँ ना, ओके देखो ना।
अनलॉक करो अभी चेक हो जाएगा।
देख लीजिएगा अभी, मैं अनम्यूट कर रही हूँ। कैसे होगा ये? यहाँ से ना ये रिकॉर्डिंग भी स्टार्ट करनी होगी? नहीं ना?
ठीक है, हो गया।
हेलो हेलो, hello.
Yes, ma'am.
Yeah, am I audible to everyone?
Yes, ma'am, you are audible.
Yeah.
Yes.
Like, do you guys want 5 minutes of break, or should I start? Okay, so let's have a break of 5 minutes.
मैम को एंटर करने के देखिए मैं तो समझ रही हूँ। मिनट्स का ब्रेक मांगा उन्होंने तो
15 मिनट्स के बाद मीटिंग लॉग आउट करते हैं,
अच्छा लॉक
लॉक करते हैं।
लॉक करते हैं मतलब
15 मिनट के बाद।
हाँ 4 बजे
तो क्लास कितने बजे तक चलेगी?
5 बजे तक। ऑप्शन नहीं है ना ये सब रिकॉर्ड नहीं हो रहा, रिकॉर्ड नहीं हो रहा है। बस मेक श्योर दैट दिस इज ऑफ एवरीडे।
So is roll number 3635 Alisha Jindal here now? 3635.
कहाँ चला गया?
Yeah, you're there.
she's there.
Okay. So now I think all of you are aware of the fact that you have to be there in the beginning, and after 15 minutes they lock you out. They lock everyone out. Then if somebody inadvertently gets locked out, they have to identify themselves, and only then selectively we unlock that person. So I can see 3635 has logged in. So you are able to access. Please answer if you are. Okay, you cannot speak. You can type the message. If you are there, if you can see the slides.
कोई नहीं है, सब ब्रेक पे। कब कर दो? हो गया ब्रेक तो। मीटिंग आईडी बता दो जरा सा। इसकी मीटिंग आईडी।
Hello?
हेलो
So Dr. Sneha, you can, I think, start. Is everyone here?
मैम they wanted a 5 minutes break so I'm waiting for that.
Okay. So what time did Dr. Arundhati finish?
Ma'am finished at around 3:30, 5:36.
Okay, 3:35. So another minute or so, and then you can start.
Yes ma'am.
हाँ
Right.
अटेंडीज का लिस्ट निकालो।
कहाँ से?
कहाँ से निकलता है भैया?
क्या?
अटेंडीज का लिस्ट।
एक मिनट
हाँ ये तो आ गया। नाउ स्क्रॉल इट फ्रॉम ऊपर से 3623 से स्टार्ट होता है अटेंडेंस। 23
सारे
कोई एक्स्ट्रा या डबल नाम जिसका हो
2-3 ना। ये कौन है?
हाँ अब इसको जैसे कॉलर 9 ना, इसका नाम नहीं है।
डिस्मिस करना है।
डिसमिस कॉलर 9। यस I'm going up.
Going down.
अचानक से 49M क्यों आ गया यहाँ पे? V के बाद M थोड़ी आता है।
पता नहीं।
स्टूडेंट नहीं होंगे ना फिर उस नाम से, इसमें डाल तो इसी नाम पे आएगा।
बट क्यों आ रहा है ये यहाँ पे अचानक से? ये नंबर से आता है।
ये रोल नंबर से।
नाम का कोई ये नहीं है। नहीं नहीं ये है स्टूडेंट है, आई नो दैट्स वाई। ये सुशील कौन है? कोई सुशील नहीं पता, डिसमिस करो उसको।
ये मैं हूँ।
अच्छा आप हो, तो ठीक है। अगर ऐसी मनमोहन जी ने वो लगा दिया था, आई वाज थिंकिंग कौन है। मैम चलो मैं म्यूट कर दूँगी, स्टार्ट करते हैं।
ये हमें ना करना पड़ेगा, इसमें LT भी लिखना पड़ेगा।
Hello. Hi, good evening everyone. It's still good afternoon. We're still
Good afternoon, ma'am.
yeah, we're still 15 minutes left for good afternoon, for good evening. Can I start now? Everybody's back for the lecture?
Yes.
Okay.
Yes, ma'am.
Okay, great. So let's begin. So the topic for today is mammary glands or the breast. Take care. Okay, so this image, is just to arouse your interest and let you know that, see, breastfeeding is one of the most noble job that can be done by a mother. Okay, this is, and it's a pictorial depiction, where it is depicting as this tree of life. So a mother is feeding a baby, the roots, the nutrition from the mother is passing on to the baby, and that's how a baby receives his first nourishments. So that is the importance of the mammary glands. So I've just got this quote. It's written by, she's a lactational consultant, and she's written very beautifully over here, that breastfeeding is a mother's gift to herself, her baby, and the earth. So these will be the objectives that we'll be covering in today's lecture. We'll be having a brief introduction. I'll be describing you the adult female breast or the mammary glands under the following points: extent, mammary bed, retromammary space, axillary tail of Spence. We'll see the features of the skin, that is the nipple and the areola, the architecture of the gland and its clinical importance. Further on, we'll be reading about the arterial and the venous supply, the nerve supply, lymphatic drainage. I'll be emphasizing few points on breast cancer, the embryological anomalies, male breast as well as male breast cancer. See, the importance of the mammary gland, it's more of a clinical topic. You will be, when you reach in your final years, surgical classes in every place, it is that time when you will understand what is the importance that we're starting today. Okay, so let me introduce you to the Latin word for the breast is mammae, and these are the most prominent superficial structures in the anterior thoracic wall, especially in the females. It consists of glandular and supporting fibrous tissues embedded within fatty matrix, along with blood vessels, lymphatics and nerves. It's of course present in both sexes. In males and in immature females, they are of similar structures. The breast, they start developing once you reach the puberty. A female reaches the puberty. They also form the secondary sexual characters of female. Source of nutrition for the neonate, as I mentioned earlier, for breastfeeding. Its shape and size may vary, okay, and that depends upon genetics, racial, dietary, age, parity and the menopausal status. Okay, so what is the adult female breast or mammary glands? These are actually modified sweat glands, okay, and they lie in the superficial fascia of the pectoral region. So more so, mammary gland is also always being taught, with the upper limb, but it is a part of the thorax, the pectoral region. Few of the things that we are going to read today, you'll be able to understand it much clearly when you will study thorax, the various arteries, the branches and the nerves, because you have not read those words till now. So just try to understand, make some concept, and I'll be happy to clear doubts at the end of the class, because I know a lot of doubts are going to be arising. Okay? So now we see the extent of the breast. So see, breast itself is a pendulous structure. That's, it's pendulous. So the base of the breast, that is its attached surface, extends vertically from the 2nd to 7th rib. So over here, this is a diagram, like this is an image of a female breast. So the vertical extension is from the 2nd rib. So see, this is the clavicle, this is the 1st rib, this 2nd rib. 2nd, 3rd, 4th, 5th and the 6th rib. So the vertical extension is from the 2nd to 6th rib. And the transverse plane is from the sternal edge medially to the mid-axillary line, laterally. So that is how you describe the extent of the base of the breast. Okay, so now we'll understand what is a mammary bed. See, we understood that the base of the breast is the one that is fixed. So this is called as mammary bed, and it is fairly constant. You know, see, the fatty content varies with the physiological process. Lactating mother, a pregnant female, it increases, decreases back again. But the base of the breast is always fairly constant, and it rests upon. So we have read about pectoralis major muscle, serratus anterior muscle, and I think you have read about external oblique, or it may be new, but you will understand, read more about it in your further lectures. So superiorly, the mammary bed lies upon the pectoral fascia. So that is the fascia which is covering the pectoralis major muscle, okay, which in turn overlies pectoralis major. So see, since breast has a huge extent, medially from the lateral angle of the sternum to the mid axillary line, so the medial 2/3 of the breast lies on pectoralis major, and the lateral 1/3 of the breast lies on serratus anterior. Inferiorly, it lies over external oblique and its aponeurosis. So external oblique, we've seen the sternum. The external oblique fibers arises, they attach on the xiphisternum. So see over here, this image I'll try to explain more. This is a much clearer image. See over here, this is the pectoralis major muscle. So that is forming the medial 2/3, and the lateral 1/3 is forming on the serratus anterior, and these are the fibers of the external oblique, interdigitating kind of, that you can understand here. Take care. Now we'll come to another terminology, that is retromammary or submammary space. By the word also we can understand, retro means something that is behind the mammary gland. Sub also means something that is under. Take care. So between the base of the gland and the pectoral fascia. So the mammary gland was lying, the bed was formed by the pectoral fascia, and underneath was the pectoralis major muscle. So the space between the base of the gland and the pectoral fascia is called as the retromammary or the submammary space. It contains loose connective tissue. The important point that you have to understand is that it is a potential space. It is not present, but it is a potential space, and it is this space that we use, if you would have been in a dissection hall, if you would have dissected the mammary glands, so it is this space that we use to separate the mammary glands. I'll show you in the videos also, so maybe that time it will be much clearer to you guys. Okay, and also since see, it is containing loose connective tissue, it allows breast some degree of movement on the pectoral fascia. So what is the clinical importance? I'll try to incorporate the clinical importance as and when along I can, so that at the last it doesn't become a lot of clinical applied knowledge to you people. So the clinical importance would be, see, if this space is suppose invaded by the cells of the carcinogenic cells, take it, the breast carcinoma. So what happens? The gland will get fixed to the pectoralis major. The space is going to be lost. So that is one of the signs that the breast cancer has advanced. It is not just in the gland, it is moved beyond the space of the gland and is infiltrating the muscles. Okay, so the next point is axillary process or tail of Spence. These are all little terminologies that you need to understand. So the superolateral quadrant. So like when you talk about the breast, even in surgical, your postings also you'll see that we'll always talk about the breast in quadrants. So see, we have tried to show you here, like we divided, it's a vertical and a horizontal line. So this suppose becomes a medial, of course, because this is sternum, so it is medial. It is medial. So this is superomedial or upper medial. Just hold on for a moment, some problem is here.
Now it's fine.
कहाँ है?
मैम का फोन आया.
अरे तो कोई है ही नहीं अकरम नाम से. खुद ही देखो ना. ऐसे बीच बीच में मत रुको ना, फिर मेरा पूरा फ्लो टूट जा रहा है.
देखो मैसेज है, मैम का कॉल आया. मैम का नंबर तो बोलो.
पता नहीं, छोड़ दो. मुझे तो इसमें कोई नहीं दिख रहा.
लॉक करो.
लॉक किया? मीटिंग लॉक किया?
हाँ वो तो कुछ नहीं करा आपने लॉक.
Ma'am, we can't hear you. Audio has gone.
Ma'am, I'm not speaking right now. I think some people have, joined the meeting, so we are trying to unlock the meeting, ma'am. कहाँ से ये?
No, I'll do that. You don't bother. You continue talking. Okay?
Yes,
Dr.
ma'am.
Sneha?
Yes, ma'am. Okay, ma'am.
We'll unlock it. I'll deal with it.
Okay, ma'am.
You just continue your
Okay.
lecture.
Okay, ma'am. Okay. So we were talking about the quadrants. So I was trying to explain, see, when it is much easier, you understand, like, of course, you've seen the extent of the breast. So, it's a huge organ. So just to pinpoint anything, you cannot say it is present above, below, side mein, right, medial. So what happens, it is a common terminology that is being used, and we divide it into 4 quadrants. Okay. So the 4 quadrants will be if it is medial, so of course these both will be the medial quadrants, and this will be the superomedial or upper medial, inferomedial or the lower medial. You will find these terms interchangeably used. Okay. Similar for the lateral quadrants. This is the superolateral quadrant or the upper lateral quadrant, and this is the inferolateral quadrant or the lower lateral quadrant. So what we are trying to say is the axillary tail of the axillary process of the tail of Spence, it is projecting from the superolateral quadrant. So see, this is the superolateral quadrant over here. So it is projecting from here towards the axilla. Okay, and the direction is upwards and laterally, and it is along the inferolateral edge of the pectoralis major muscle. So now we know the anatomy of axilla. We know how the pectoralis major muscles are twisting and then inserting. So we can understand this. Okay, and it enters the axilla through an opening in the axillary fascia called as the foramen of Langer. So sometimes you can be asked, and this can also be seen in MCQs, even in your post-graduation exams also you'll find that foramen of Langer, like what is it? So you need to just know this term, that it is a opening in the axillary fascia through which the axillary tail of Spence passes. Okay? And they extend through the deep fascia up to the apex of the axilla. The importance of this axillary tail of Spence lies at the point.
Just to point out to the students, this is an extremely important relationship, that there is actually a projection of the parenchyma of the mammary gland into the axilla. This has grave implications in breast cancer. So it's very important to know about this. That's why it's being stressed. Please continue, Dr. Sneha.
Yes, ma'am. So I was talking about, because the most important, as ma'am just mentioned right now, so breast cancer is, it has a great implication on the breast when you read about the breast cancer, because it comes in contact with the anterior group of the axillary lymph nodes. So over here, see over here also, in this image also, see, you can see that this is the axillary process, see, and this is the pectoralis major muscle. So how it is twisting and following around the inferolateral angle of the pectoralis major muscle, and it is coming in contact with the anterior group of the axillary lymph nodes. So the clinical importance over here lies that when this is enlarged, may be mistaken as a tumor, since it's a breast tissue. So it is going to have some hormone receptors also. So with your menstrual cycles, with pregnancy and everything, of course, they will enlarge. So this can be mistaken as tumor or a lipoma or as enlarged lymph nodes. So whenever you're palpating clinically the lymph nodes and everything, you need to have this in mind, that in females, the extension of the breast tissue is up till the axilla. Okay, so now we'll study the features in the skin. So you have a nipple, which is a very prominent feature, which you can see. It is small and surrounded by a colored area of the skin called as areola. And it projects just below the center of the breast anteriorly. Okay? Just below the center, not in the center. And of course, this is all variable. See, with the parity, with age, with genes, these things changes. It may be cylindrical, rounded, hemispherical or flattened. And few of the factors are which decide these shapes and sizes are the effect of the developmental, nervous, hormonal factors or external temperature. What is the level of the nipple? See, in males it is fairly constant. It lies at around the 4th intercostal space along the mid-clavicular line. But in females, of course, this will be changing with the size and the shape of the breast. But in generally, if you can say, it overlies the 4th intercostal space in most young women. The importance of the nipple lies in the fact that it is pierced by 15 to 20 lactiferous ducts. We'll be seeing in further consequences slides why am I emphasizing over here that it is being pierced by 15 to 20 lactiferous ducts. It contains smooth muscle cells, both circular as well as longitudinal. And the importance is the circular smooth muscle cells helps in the erection, that is important for the suckling and the latching of the newborn to the mother for the breastfeeding, and longitudinal, they retract the nipple. One more important point which I want to emphasize is the nipple is richly supplied by nerves, so it is highly sensitive area. Okay, areola. It stands for a small area. So that is the colored area surrounding the nipple. It is pigmented and circular area of the skin around the base of the nipple. The outer margin have modified sebaceous glands which are enlarged during pregnancy, and it is called as tubercles of Montgomery. Since it is sebaceous glands, it is going to have oily secretions. So what is the function of this oily secretions from the areola? It acts as a protective lubricant. Also, it facilitates in latching of the newborn for the feeding. It also contains few sweat glands, some accessory mammary glands, and when these glands are enlarged during the pregnancy, they are called as subcutaneous tubercles. You just need to understand these words, so that's why I've highlighted the subcutaneous tubercles. So you know that they are just enlarged in the pregnancy. In a normal female, you will not find that. Okay? The skin of the nipple and areola is rich in melanocytes. Since it is pigmented, so it has a lot of melanocytes. And a point to understand over here is the number of melanocytes increases with pregnancy. And this darkening of the skin with the pregnancy is irreversible. So once a female becomes pregnant and the color changes of the areola, it cannot be back to her pre-pregnant status. Okay, and beneath the areola, each lactiferous ducts, so these lactiferous ducts I emphasized earlier, the 15 to 20 lactiferous ducts which were piercing the nipple. So beneath the areola, each lactiferous ducts dilates to form a lactiferous sinus. So see what you understand by duct and a sinus. A duct is of course a tube, just to make you understand. Of course, when you read more further, you'll understand duct is different, and sinus is a dilated portion. So over here, see, you can see over here. I'll just try to show in this image. See, this is the nipple, this is the areola, and see, you can see these small sac-like dilatations. So these are the lactiferous ducts. Okay, and these lactiferous ducts, they lined with the spiral arrangement of, they have contractile myoepithelial cells. So this also you will understand that why contractile myoepithelial cells, that is for the ejection of the milk when the baby is suckling. So they require the myoepithelial cells will contract, and thus the milk will pass from the breast to the baby. Something that you need to understand now is the architecture of the gland. See, the breast is composed of acini, which is a smaller unit. Again, a number of acini are going to form lobules, and number of lobules is going to form lobes. See, if I would like to show it, see these small grape-like bunches, these are the acini. Okay, this is a lobule, okay, and these are lobes. So each lobe, see, is having its own lactiferous ducts. So we can say that if there are 15 to 20 lactiferous ducts which are piercing and reaching the nipple, so there are around 15 to 20 lobes. Okay, you are able to understand this. So see, these lobes, they are arranged in a radiating fashion, like a spoke of a wheel, and converging on the nipple. Each lobe is having its own lactiferous ducts. Okay. So if you see the breast components, so there are three components. Three components of the breast are there. It has a glandular tissue, okay? The glandular tissue, the glandular part. It has fibrous septae, the fibrous part, and it has the intralobar fatty tissue. So you have the glandular part, you have the fibrous septae as well as the fat. So these three things, the three components constitute the breast. Okay, so we'll understand more, just a little further points. The glandular tissue. So the type of gland, is tubulo-alveolar type. So tubulo-alveolar, just understand. I know even if you can't understand and these concepts, we'll be teaching you more further when you'll have the classes on the glands, exocrine glands. So this is just a tubular, see, it's a alveolar, multiple branched alveolar, and this is a tube along with the alveolar. So this is the type of variety of the gland that is found in the mammary glands. The fibrous tissue, it supports the lobes, as we can see even in the previous image and even in this image. See, the fibrous tissue is going to support the lobes. So that is the importance of the fibrous tissue. They help in anchoring the parenchyma, okay, to the skin and the underlying pectoral fascia. These fibrous septae are called as suspensory ligament of Cooper. This is also another name that you need to know, suspensory ligament of Cooper. They help in maintaining the protuberance of a young breast. That is, in young females, you will see the breast are protuberant. But as with the age and with the age progresses, because of the atrophy, it becomes pendulous. And the intralobar fatty tissue, it gives the round contour to the organ. And just a point to understand is that this fatty tissue is absent beneath areola and nipple. So see over here there is no, even if you see that there is scanty over here, just beneath the areola. And this is in abundance in the rest of the gland. This is also just an image just to tell you that how a branched and tubular compound gland will look like. So the clinical importance over here is these fibrous septa. Yes,
just pause a minute here, Dr. Sneha.
Hanji, ma'am.
This will be done in detail in histology, but it's a good thing that Dr. Sneha has just introduced these pictures to explain to you what it means in the context of histologically visible glands. So there are different types of glands. It may be like a straight tube, or it can be a branched tube, or it can be a coiled tube, or it can be several branching compound tubular glands, or if it has a sac-like structure at its end, then it is called a tubulo-alveolar gland. So alveolus, the term refers to a dilated sac-like structure which forms the secretory part of some glands. So the secretory part may be tubular or it may be sac-like. So based on that, this classification is made. Please continue.
Yes, ma'am. So, we read about the word suspensory ligament of Cooper. So, what is the clinical importance? That is the growth. Like, suppose if the breast cancer cells are extended along these ligaments, so they are going to produce dimples. See, something which is already taut. Okay, taut, T-A-U-T, taut, as in it's tight, and suddenly some cells are there and it's contracting. So, of course, it is going to produce dimples. It will be pulled. So, you will see dimples over the smooth skin of the breast, overlying skin. Or if it is infiltrating the retromammary space and reaching up till the pectoralis major, then this will cause the fixation to the pectoralis major, both being signs of advanced breast carcinoma. Okay, now this is just some concept that I want to introduce you. See, we have read about the lactiferous ducts. We read that they are 15 to 20, and they have lactiferous ducts. Okay, so they are arranged in radiating manner, converge towards the areola. At the level of the areola, each duct dilates to form a lactiferous sinus, which we saw in the previous image. So the importance of lactiferous sinus is this acts as a reservoir of milk, which finally then opens onto the nipple. So this is the way forward from the lactiferous ducts. Now if we move way backwards for the lactiferous ducts, so see, each lobe was having one lactiferous duct, but the lobe was constituting many lobules. So each lobule is going to have the segmental system. So the segmental ducts will be in the level of the lobules. Okay? And these segmental ducts are then further going to be finally divided into terminal ducts. So the acini are going to have the terminal ducts, which terminal ducts are finally then going to open into the acini, which are the grape-like clusters. So the importance over here lies is, why did I emphasize so much on making you understand what is a lobe, what is a lobule, what is an acini, how is the duct moving forward and how is the duct moving backwards from the lactiferous duct is because the area of the breast parenchyma drained by one terminal duct is known as a lobule. Okay, and the terminal duct lobular unit, like this unit is called as the functional milk secretory component. So it can be asked to you in vivas or in MCQs that what is the functional milk secretory component of the breast. So it is the terminal duct lobular unit. In this image also we can see these are the lactiferous ducts. This is the lactiferous sinus. See, this is a lobe of a mammary gland. So each lobe is having its own lactiferous duct, which is then further dividing into segmental and then into terminal lobules, and this is, see, this is a lobule. So this is the functional unit of the mammary gland, and this is a terminal duct. Okay, so is everybody clear? Hello, are you guys able to understand and cope up with what I'm trying to tell you and make you understand?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay, thank you so much. Okay, sure. So this is just an image. This is just to tell you and show you that, so this is how, if we see the breast anterior view. So see, this was the nipple. These are the lactiferous ducts. Okay, lactiferous ducts again. These are the suspensory ligaments, the fatty tissue, the glandular tissue, and this is how the breast will look. This is just a schematic diagram. Of course, this is not the exactly dissected specimen. And this is the sagittal section. So over here also we can see the lactiferous sinuses, the dilated ones, the interlobular fat, the lactiferous ducts, the septae, that is the suspensory ligament of the Cooper, these, and then this is the retromammary space, which is a potential space. Okay, and then this is the pectoralis major muscle on which the breast is lying. The medial 2/3 of the breast is lying. The lateral 1/3 is on the serratus interior, and inferiorly it is on external oblique and its aponeurosis. So see, when I'm emphasizing so much upon ducts, so there has to be some clinical importance. Everything that you're learning and understanding is going to be implied in your clinics. So let's just study something about that. See, the different portions of the duct system, they're associated with different kind of diseases that you'll come across when you'll be posted in surgery. So the larger ducts, okay, they are the site for ductal papilloma and duct ectasia. The dilated smaller ducts are the site of fibroadenoma. That is a condition that is during the development of the breast. Cyst formation and sclerosing adenosis. The terminal ducts, which were forming the part, the lobule, is the site for the majority of the breast carcinomas. So this is just to tell you the importance that why do we need to know so much about the ducts and the lobules. Let's now understand the arterial supply of the breast. See, why would an arterial supply of breast be very important? See, because breast reconstruction, plastic surgery, everything, it all relies on the pedicle. You know, when you transplant, when you construct something, so you need a pedicle, you need the graft to give a blood supply. So that is why you need to understand the arterial supply of the blood, which way to give the incision so that you have the least scar. See, breast has a lot of cosmetic reasons. So the arterial supply of breast is important in that context. See, we have already read about the axillary artery. So whenever an organ is present, it will of course derive an arterial supply from the nearby area, the nearby blood vessels. So see over here, this is the axillary artery. So it is of course going to get some branches from the supply from the axillary artery. Then over here in the midline, over here this is the internal thoracic artery. So this is a branch of a subclavian artery. So it is going to derive its blood supply, some from the branches of the internal thoracic artery. Some from these you'll understand more when you read thorax. These are the posterior intercostal arteries. So these posterior intercostal arteries will also give the branches. Then the anterior intercostal arteries will also give the branches. So that's how you will understand how the blood supply of the breast is there. So just to enumerate further points, the axillary artery is then via the superior thoracic artery, the pectoral branch of the thoracoacromial artery, and the lateral thoracic artery. You have read all these arteries, you have also seen it. Okay? And yesterday in the class also, ma'am really taught you very well, you should be able to understand it. Then you have the internal thoracic artery. So it is going to give the perforating branches to the anteromedial. See, again the same words, superomedial, anteromedial. This is how the clinician is always going to emphasize when you ask, suppose when you asked to write a short note on blood supply of breast. So what you simply have to do is you have to draw a schematic diagram, divide the breast into 4 quadrants, and then write that the superomedial quadrant is supplied by these branches, the infromedial is by this branches. So that's how you will also understand, and the concept will be also clear to you. The 2nd to the 4th anterior intercostal arteries also supply the perforating branches. I want to emphasize over here one point, that the supply of the nipple is basically by the 2nd perforating artery. It is the largest blood supply, and it supplies the upper region of the breast, the nipple and the areola, and also the adjacent breast tissue, and of course the posterior intercostal arteries which are here. The venous drainage always coincides with that of the arterial supply. So the venous drainages of course will be again by it is a circular venous plexus around the areola, and then it drains from the areola backwards by axillary vein. So over here we are having, this is the axillary vein, the internal thoracic vein, and the intercostal veins, the posterior as well as the anterior intercostal veins. See the importance of like the blood supply and the venous drainage lies in the fact that any carcinoma, I'm not just talking about on a specific term, it has two routes of spread. You know, the two routes of the spread will be the hematogenous, that is through the blood, both arterial and the venous, and through the lymphatic drainage. So over here, if you can see this, the breast is receiving such an extensive arterial as well as the venous supply. So this is of course going to lead to metastasis to so many other places. So through the intercostal veins, these intercostal veins, the both anterior and posterior, they reach the azygous veins, and from here they can communicate with the intervertebral venous plexus of Batson. So that this vertebral venous plexus lies in the vertebral column. So that's how the metastasis of a breast can reach to a vertebral column, and a patient presenting with a symptom could be just backache, a severe backache. And when you examine and further investigate, and you'll understand, you'll see that the breast cancer is already metastasized and it has reached to the vertebral column, vertebral bones. It can also reach to the intracranial, sagittal and transverse sinuses through these venous drainage. So now intracranial, sagittal and transverse sinuses lie in the dura. Okay? So that is how it is reaching up to the central nervous system. It can also establish venous communications with the cervical vertebra, clavicle and humerus. This hematogenous spread of the breast cancer is by the retrograde venous flow. The walls become incompetent of the veins, and it allows the cancer cells to pass backward. Now moving further to the nerve supply. So the nerve supply of the breast is by the anterior and lateral cutaneous branches of the 4th to 6th intercostal nerves. So we have read a typical spinal nerve. We know how the anterior, the lateral cutaneous branches are formed. So I not need to go into that. You just need to understand that these are from the 4th to the 6th intercostal nerves. These nerves carry both sensory fibers from the skin of the breast. Okay? I told you that the nipple is the most sensitive area. So they are going to carry the sensory fibers from the skin of the breast, as well as the sympathetic fibers to the blood vessels in the breast and the smooth muscles. I told you that there are two smooth muscles, circular and the longitudinal, for latching and as well as for the retraction of the nipple. So these are the anterior branches, the anterior cutaneous branches, and the anterior and posterior of the branches of the lateral cutaneous nerves. See, I need to emphasize more upon this, that the nipple is supplied from the anterior branch of the lateral cutaneous branch of T4, okay, which forms an extensive plexus within the nipple. The importance over here lies that it has sensory fibers in forms of free nerve endings, Meissner's corpuscles and Merkel's disc. You'll read about them more further, that free nerve endings are basically what? They're nociceptive, the pain is from the free nerve endings. The Meissner's corpuscles is for the fine touch, and Merkel's disc is for the pressure. So these are the senses that you can understand. Then that's why I just mentioned that nipple is highly sensitive area. It is a tremendous amount of nerve supply along with these mechanoreceptors, the free nerve endings, the Meissner's corpuscles and the Merkel discs. These receptors are essential in signaling the suckling. See, when a baby is being latched to a breast, so of course the suckling process is going to start, and how the lactiferous sinuses, which are going to store the milk, is going to pass. So all these sensations is for the process of suckling. And the secretory activity of the gland is largely controlled by the hormones, both ovarian hormones as well as the hypophyseal hormones. Okay. So now moving further, we have read about the arterial supply, the venous supply, the nerve supply, and lastly the lymphatic drainage. See, I cannot emphasize more and more that why lymphatic drainage is so important of the breast. You get a question almost like every alternate year upon this, and it is very important, both as well as from the anatomical point of view as well as from the clinical point of view. So I'll just try to make you understand how the lymphatic drainage is important. See, the great clinical significance lies because of the metastasis that I already told you about. The breasts are the site of malignant changes in as many as 1 in 8 women. That is worldwide. This is the worldwide data. And according to American Institute of Research Cancer Research, breast cancer is the most commonly occurring cancer in women and the second most common cancer overall. So this is just some numbers for the most common cancer it is also most common cancer in women in India. So over here you can see this pink color, the bright pink color is depicting the breast cancer, and just see the incidence is so high, even the breast cancer incidence is high, even the mortality rate is so high with the breast cancer, and thus lie the importance of understanding the anatomy of breast and its clinical importance. And one woman is diagnosed with breast cancer in India every 4 minutes, and every 8 minutes a woman is dying of breast cancer. So that's where the importance of the lymphatic drainage of the breast cancer lies. Okay, to understand the lymphatic drainage of the breast is by two processes, like the two components of the lymphatic drainage. One, it is draining the parenchyma of the breast, including the areola and the nipple. Okay? And the other one will be the draining of the skin, excluding the areola and the nipple. Okay. So what happens? See, we just read that everything from the nipple is radiating and it is going back. So lymph follows the venous drainage. So that is also a subareolar plexus of Sapi is a circular, radially circular area from where the lymphatic is being drained, and then it moves backward. So most of the lymph, see, the nearest of the lymph node is the axillary lymph node. So most of the lymph, that is the 75% of the lymph, drains into the axillary nodes. And we mentioned, the axillary tail of Spence lies near the anterior group of the axillary lymph node. So it is the anterior group which receives the most of the lymphatic drainage. Some can also go to the posterior group. And from this anterior and posterior group, they then go to the central group of the lymph nodes and then to the apical group of the lymph nodes. The rest 20% drains into the parasternal or the internal mammary nodes. Okay? And the remaining 5% is draining into the posterior intercostal lymph node. So it is just following the venous system, if you can see. See, the most 75% is following the axillary, the 20 the internal mammary or the parasternal lymph nodes, and the remaining 5 are the posterior intercostal lymph nodes. So we can just see the pectoral, the humeral, and the subscapular lymph nodes. So the pectoral is the anterior group of lymph nodes, and the posterior, the subscapular, they are going to drain into the central group, from central to apical, and from apical they are going to be reaching the subclavicular lymph nodes. Okay, so this is the drainage of lymph from the parenchyma of the breast, including areola and nipple. So this is also a schematic diagram. Over here also you can see how these radially spooking out. Okay, this is the axillary, the internal mammary, and the posterior. That's not shown in this diagram, but the posterior are going to lie here, the posterior intercostal group of lymph nodes. That is just the 5%.
Okay. Excuse me, ma'am.
Yeah.
How is lymphatic drainage related to cancer, related to breast cancer? When there are any neoplasia, like suppose if there is any abnormal growth of the cell, that is called as cancer. So what happens? These cells, they detach. They're very rapidly growing cells, and then they move forward. You can see the seeds of the cancer cells are going to be traveling. They take two routes, either hematogenous, either from the blood supply or from the lymphatic drainage. So it is this, you'll understand all these processes when you move into pathology. The topic today is covering almost pathology, surgery, many other things. So it is not just related strictly to anatomy. So you'll understand how these neoplastic cells are, traveling taking the lymphatic route and, finally reaching other places and doing the metastasis. And so the parenchyma becomes cancerous or what? And that will be metastatic.
The cells. Either the cell lining the ducts, the cells found in the parenchyma, they all become cancerous.
Okay, and they metastasize and move to other parts of the body. So that's what you're meaning.
So you'll understand that the primary cancer is the breast cancer, but the patient is presenting with a secondary metastasis that is into the vertebral column or in the dural venous sinuses. So you understood now?
Okay, I get it. Thank you, ma'am.
Okay, welcome. ठीक है. So now the second part of the lymphatic drainage is the draining the overlying skin excluding areola and nipple. ठीक है. So here also from the outer part of the breast, the outer part of the skin of the breast drains into axillary. From the upper part it is the supraclavicular, because of course they are the nearest. Then they find everything that is near to it. ठीक है. So the outer part is close to the axillary lymph nodes, upper part is close to the supraclavicular lymph nodes, the medial or the inner part is close to the parasternal or the internal mammary lymph nodes, and the lower part is close to the rectus sheath. We read about external oblique aponeurosis, external oblique and its aponeurosis. So you will understand this further, that external oblique forms, the aponeurosis forms the rectus sheath, which is, in the center of the abdomen, linea. I cannot go into further, that is an abdomen topic, but just please understand this, that the lower part is going to the rectus sheath lymphatics. I'll show you in the video also, we have a video where you can understand these points. So the point of importance that I want to tell you, see, the cancer cells are there in the breast, but what are routes they're taking and reaching where and where, that through the lower part, it is communicating with the rectus sheath. From here they are reaching the subperitoneal plexus of the lymph nodes. Peritoneum ke niche is the subperitoneal plexus. From there they are draining into the subdiaphragmatic nodes. Diaphragm, peritoneum, everything is lying like below the diaphragm. Okay, thorax, diaphragm divides into thorax and abdomen roughly. So they are reaching the diaphragmatic nodes, and from here they are reaching to the falciform ligament. Falciform ligament is found in the liver, that anterior line that you see in a liver, if you've seen the image of a liver, to reach the bile duct. And what happens here is, the clinical importance lie here is that a patient can be presenting with you to the obstructive jaundice. A patient comes with the jaundice and says that, he has a long history of yellow urine and white stools and everything. And you are going to find the primary in the breast cancer. When you're going to do proper investigations and see, then you'll understand how the metastasis of breast is reaching up till the bile duct. And the presenting symptom of the patient is something else, but the primary cause of the symptom is breast cancer. Okay, am I clear? Are you understanding? Hello? Hello? Hello?
Yes, ma'am. Yes, ma'am. We are able.
You are able to understand. I'm just trying to tell you that how a breast cancer is going to spread at so many places, and that is why it is one of the most common cancers. Okay. Also from the subperitoneal plexus, it is going to drop in the peritoneal cavity, by the gravity, because of the gravity. And this causes transcoelomic migration. Okay? And because of this dropping of the cavity and transcoelomic migration, you'll understand, that ovary is also found in the coelom. You'll understand this process. They are going to get deposited on the ovary. So how a breast cancer, which is a primary breast cancer, causing a cancer of the ovary. So this is called as Krukenberg's tumor. These are some fancy name given, and you just need to understand, because the moment you say Krukenberg's tumor, everybody is going to understand. Every clinician will understand that what is a Krukenberg tumor. The cutaneous lymphatics, that is this lymphatic that we just talked about, the second part. It also communicates across the midline to those of the opposite breast. Okay? So that is how a breast disease which is unilateral can, because of the help of the cutaneous lymphatics, will be bilateral. Okay? So that is where lies the importance of the cutaneous lymphatics. In this image, I am just trying to show you that, the lymphatic and the venous drainage, they just follow each other. The direction of the lymph flow from the breast is parallel to that of the venous drainage. Okay. So now we'll read some clinical anatomy in context of the breast cancer. We have already seen this further. This is just to, just to summarize that if there is infiltration of cells in the suspensory ligament of Cooper, so which was the fibrous septae. So what was happening? This was causing the dimples on the overlying skin and fixation to the pectoralis major muscle. If there is cancerous growth along the lactiferous ducts, this was leading to retraction of the nipples, because these ducts were then finally opening onto the nipples. If there is obstruction of cutaneous lymphatics, which I just mentioned, okay, so what happens is, firstly, it is going to, a unilateral disease can become bilateral. Secondly, you can also find a manifestation of stagnation of the lymph. and edema of the skin around the hair follicles of the breast. This condition is called as peau de orang. Okay, so I'll just try to show you, like this is an image over here. I've just tried to show you the skin, the breast skin becomes like that of an orange. It look an orange peel because these small hair follicles over here, these the hair follicle pits, there is edema here and there is stagnation of lymph. So this is called as peau de orang. Okay, then let's moving on to applied anatomy. See, these are few changes in the breast that you can see, you know, the branching. Of course, the young female breast, before the puberty, there is no lactiferous ducts, there is no branching. Okay, but at as the female reaches puberty, there is branching. The branching pattern also changes with that of the menstrual cycle in the first half, in the second half. Then when the female becomes pregnant, lactating of course will have milk production. When after cessation of the lactation, and see at the breast of the elderly woman, they are usually very small, and that is because of the fat deposit over here, the fat that was constituent of the breast is decreased, and there is atrophy also of the glandular tissue. So these were providing the contour to the breast. So these, with the atrophy, it is getting small in size. I'll just introduce a term here that is called colostrum. I think maybe you must have heard colostrum. That is the first milk. It is a creamy white to yellowish pre-milk fluid, you know, which is secreted during the initial episodes of the lactation, and it is believed to be rich in proteins, immunoagents and growth factors, and it is very important for the nutrients. It is rich in nutrients and is important for the infant. One thing that you all see is that is witch's milk in the newborn. You know, some in pediatrics and all, you can see this, or you know, a mother coming with a complaint that, you know, the baby was just born few days back, and you know, there is secretion of milk from the nipples. So you need to understand and make the mother, if you'll understand, if you know, then you'll understand that this is normal. That is because when the baby is in the uterus, so there is you know, the maternal and the placental hormones are crossing the the placental barrier, and they are causing proliferations of the ductal epithelium in the fetus's breast. So this proliferation may cause then the swelling of the breast, and it can be present in both sexes, like a male baby as well as a female baby, and this is for the first week of the life. And in some cases, the presentation can also be that the milky like fluid is coming out from the, you know, the newborn infant's nipples. So you need to understand and make the mother understand that this is normal, and after 1 week this will go away. And this condition resolves spontaneously as the maternal hormone level will fall in the newborn. Okay, again I have already mentioned about these you know, breast quadrants, but then this need to be emphasized again and again because this is really important. So for anatomical location and description of tumors and cysts, the breast is divided into 4 components. Over here it is just showing the percentage of the malignant tumors that you find in each breast. Okay, so we can move forward. Okay, we'll just talk briefly about breast cancer. It's a huge humongous topic, and you have whole chapters and lectures when you are reaching in your postings and in your final years. But I'll just introduce you because since we are studying about mammary gland and the female breast, so you need to know about it. These are malignant tumors, usually adenocarcinoma. Adenocarcinoma means that of the glandular tissue. So because since breast has so much of glandular tissue, so then adenocarcinoma of the breast is common, arising from the epithelial cells of the lactiferous ducts in the mammary gland lobules. The spread by the means of the lymphatic vessels. So over here, if somebody has just asked the question, you know, how lymphatics? So the spread by the means of the lymphatics is the most common, called as lymphogenic metastasis. There is abundant communication among the lymphatic pathways. We just read, you know, that how lymphatic pathways are, you know, through axillary nodes, parasternal nodes, posterior intercostal nodes, the cutaneous lymphatics. So that is the reason why there is abundance of, you know, huge metastasis that can be seen in the patients of breast cancer. The most common site being the axillary lymph nodes. Why? Because of course 75% of the breast parenchyma was being drained by the axillary lymph nodes. Okay. So then the invasion of the retromammary space, which I told you, you know, it's a potential space lying upon the pectoralis fascia. It causes the breast elevates when the pectoralis muscle will contract. So you have to understand where, you know, you ask the patient to, you know, the muscle, you ask the patient to elevate the, you know, to function of the pectoralis major muscle, and you see that along with the action, the breast is also elevating. So this movement is a clinical sign for you to understand that the breast cancer has invaded from the parenchyma and up to the muscles. And we read about this also, you know, through the posterior intercostal veins from the azygous and hemiazygous venous system is going to lead to the, you know, metastasis into the vertebral bodies, the cranium, the brain, and by the internal vertebral venous plexus of Batson to the spinal cord. So this is just an image to show you that, you know, this is how a bloody discharge from a breast carcinoma will look like. Just few investigations, you know, if a patient comes with a breast symptom, you can do mammography, ultrasound, MRI, you can do FNAC, that is, you know, fine needle aspiration cytology to see the cell types, whether it is a glandular, it is an epithelial cell, what kind, and then finally it is triple assessment. So triple assessment is the most important point because it gives you almost a confident diagnosis of 99.9%. How do you assess? You use the clinical you know, the clinical knowledge, that is the age and the examination. You do imaging, that is ultrasonography or mammography, and you do pathology, FNAC or a core cut. Core cut means you're going to take a part of the breast tissue and you're going to make slides of it, section it and slide and see it on the slide and then study the cell cytology. So combining of all these three, clinical, imaging and pathology, is almost going to give you a confident diagnosis of the type of the breast cancer. Sentinel node.
What is age gonna do in breast cancer?
Sorry, what is?
What is age gonna do in case of breast cancer? How is that?
See, age is going to be, yeah, age is an important factor. See, age is an important factor because with the current lifestyle that we're having, the increase of breast cancer in younger women has increased in the recent, in the last decade. So earlier it was considered to be just a carcinoma of an older woman, you know, somebody who is above 50, has given birth to babies and has in a postmenopausal phase. But with current trends and the lifestyle changes that we are living, it is very common now, very commonly now seen in, you know, in adult age group, in younger age group also.
Okay, ma'am.
Okay, sentinel node biopsy. This is an important topic, asked many a times, can come as a short note, and you need to just understand the concept that what is a sentinel node biopsy. A sentinel node is defined as the first lymph node that is draining the tumor bearing area of the breast. So see, a tumor bearing of the breast can be a upper quadrant, lower quadrant, upper lateral or upper medial, any of the 4 quadrants. So a sentinel node will be the one which is the first lymph node. So what happens sometimes, you know, when you have to assess the patient and you don't know that what is the extent of the metastasis or what is the extent of the breast carcinoma, and you need to know, see, because you know, as a clinician you'll be knowing that it has a rich lymphatics, so you assume that there might be already a metastasis. So to confirm your diagnosis, you need to find out that have it been, you know, has it been, you know, like moved up to the level of the lymph nodes. So to do this, what you have to do is, this technique is currently becoming a standard of care in the management of axilla in the patients with clinically node negative disease. So like suppose you have marked the patient as clinically node negative, as means there are no lymph node palpable. So such kind of patients are being given a term known as clinically node negative patients. So in them you have to do the sentinel node biopsy. What you do is, you pre-operatively you inject the dye, okay, a blue color dye is given along with a radioisotope labeled albumin to the breast. The site of the injection is subdermal plexus around the nipple. So since you know that the lymphatics of the breast was moving away from the nipple, you know, in a radial manner, I emphasize that point also, that in a radial manner it is moving away from the nipple and then to the breast parenchyma and then to the various lymph nodes. So the site of injection is subdermal plexus around the nipple, and the dye and the radioisotope labeled albumin. So what happens, because of the lymphatic drainage, this dye is going to pass and it is going to reach the lymph node. So the primary marker passes the primary node draining the area, and what happens is, this is then detected visually or with a handheld gamma camera. So see, if you are not sure, you know that the patient is having breast cancer, you know that you have to do a mastectomy, whatever kind of mastectomy you decide, and you need to, so whether it has to, you know, you have to remove the lymph nodes or not. So this can be done pre-operatively, and then when you open the patient on the OT table, you can see the lymph node which is blue. I'll just show you the image. I have put the image. See over here. See, this is how a lymph node will look like, a blue color lymph node. So you know that this is the spread has been up to this level of lymph nodes, axillary lymph nodes or your parasternal lymph nodes or posterior intercostal lymph nodes. And this, you can then just resect it, take it out, give further frozen section histological analysis to just see the type of the cancerous cells. So this helps you deciding, you performing the surgery, you perform this procedure of sentinel node biopsy, you send it for the frozen sections, and then at the moment you can just see and decide within few minutes that it is whether a ductal carcinoma or epithelial carcinoma or adenocarcinoma. So this is the importance of the sentinel node biopsy clinically. Is it understood by everyone? Because this is important and you have been asked about this. Hello? Hello? Hello?
Yes.
Okay. Okay, then you're having various treatment modalities. So the main basic principle of the treatment of breast cancer is to reduce the chance of local reoccurrence and to reduce the metastatic spread. So these are various methods that you can read it further. Surgery with or without radiotherapy, you have chemotherapy, hormone therapy and palliative therapy. Palliative therapy is one, when the spread is way beyond what you can do. It's like way beyond, you cannot resect the tissue. Nothing can be done at the point of surgical, at the level of the surgery. So you just give the symptomatic treatment to the patients. Here are a few surgical incisions. You just need to understand what is the anatomical basis of giving these surgical incisions. A radial incision is preferred in the inferior half because it is less vascular than the superior quadrant. A circumferential incision is preferred in the upper half of the breast, that is to avoid the damage to the lactiferous ducts. Incision is always tried to be made along the inferior cutaneous crease. So the inferior cutaneous crease is the crease along which the breast is hanging over the thoracic wall. So this is cosmetically beneficial because I told you earlier also that breast surgeries, it's all cosmetically related. And incisions near the areola or onto the breast itself are dilated radially. Okay? That is because you have heard about Langer lines. So these Langer lines, they run transversely, and this is again the same reason, for the cosmetic reasons. And these are just the type of mastectomies that you have. You can do the simple, the radical and the lumpectomy or the quadrantectomy. Then of course once mastectomy is done, a patient of course has a lot of a psychological problem. A patient comes with a lot of psychological problem, because of course breast being cosmetically relevant. So various breast reconstructions can be done. You can use tissue expanders, you can use silicone implants. One thing that I just wanted to mention, you've heard about latissimus dorsi, you've read this muscle. So you can now know that a flap reconstruction of breast can be done with the help of this muscle, latissimus dorsi muscle. These two words you have not read, transverse rectus abdominis muscle, this is also used. I'm just trying to show you the transverse rectus abdominis muscle is somewhere here. It's transversely lying at the abdomen, and the deep inferior epigastric perforated artery flap. So these are the three kind of flaps that are preferred for the breast reconstruction. Sorry. The embryological anomalies, of course, they can be polymastia, that is like supernumerary breast. So supernumerary breast, this is the milk line, this is a milk line. This is for the mammals, because see other mammals like the cats and the dogs, they're having so many numerous nipples. So this is the milk line. In humans we just have a single pair of breast or nipples, but there can be at times supernumerary breast called as polymastia, accessory nipples called as polythelia, micromastia, that is excessively small breast, macromastia, that is diffuse hypertrophy of the breast, and see this is polymastia. This is polymastia. Okay? See, this is supernumerary nipple. It's not gonna it's just a congenital anomaly. It's not going to cause any symptom or problem to the patient, and it can be an accidental finding when you're examining the breast for some other reason. Then this is amastia, just one breast is developed, one breast is not developed. Okay? And this is gynecomastia, that is the hypertrophy of a male breast tissue. This is both. This is unilateral gynecomastia and this is bilateral gynecomastia. You need to just understand these words, supernumerary nipples, same thing which I have mentioned earlier. along the milk line that is running from axilla to the groin, and they may or may not be associated with breast tissue. The importance lies over here is that they can be mistaken as warts or moles. So you need to understand this, that if it's a supernumerary nipple or a wart or a mole. Then you need to understand this is retracted nipples. So retracted nipples.
So Dr. Sneha, just a minute.
Hanji.
So don't worry about this part. She's mentioning it for completion. Just get the terms right now. During embryology lectures of this region, you will be studying all these anomalies. So right now, just for completion, she's mentioning their names. Please go ahead.
Yes, ma'am. So the retracted nipple can be both congenital as well as of recent occurrence. So if it's of congenital occurrence, you don't have to worry. See, this is a retracted nipple of a congenital occurrence. So there's nothing to be worried, that is just a developmental anomaly. But if the retracted nipple is of recent occurrence, and if a patient comes and tells you that, suddenly I am seeing that my nipple is retracted, then you need to be worried about this as an underlying carcinoma, which is pulling the lactiferous ducts. Okay, see, this is a retracted nipple. Again, a retracted nipple. Okay, moving on to the last topic. This is just since we have read the whole topic about the female breast, so just few points about a male breast. These are rudimentary throughout the lives. They have ducts, but there is no alveoli or lobules. There is just ductal system, which is supported by fibrofatty tissue. And the breast tissue does not extend beyond the margins of the areola. So that is just at the level, just till here is the presence of breast tissue. I just mentioned earlier also gynecomastia, that is the benign proliferation of the subareolar breast tissue in males, and this is the clinical conditions. So you just read about Klinefelter syndrome in the previous class, that's a 47XXY, the endocrine causes, some neoplastic conditions and impaired liver function. It can also be drug-induced gynecomastia can also be seen. So males have also equivalently the rich lymphatic drainage, and it is because of this rich lymphatic drainage that the prognosis of breast carcinoma of males is worse than that of the females. Why? Because see, there is no glandular tissue, there cannot be more of signs that you can see, and when a patient comes, when a male patient comes, with a breast carcinoma, it has already metastasized and almost in the late stages. So that's why the prognosis of breast cancer in male is worse than that of the females. It accounts for almost around 1% of the carcinoma. These are the probable etiological factors: radiation exposure, people going on for radiotherapy, estrogen therapy, Klinefelter syndromes, testicular feminizing syndromes, obesity, and they tend to be more advanced at the time of presentation because of the less subcutaneous fats and the high lymphatic drainage. The prognosis is very poor for a male breast carcinoma because these cells rapidly metastasize into the thorax through the small amount of intervening tissue. There is no much of fat since the tissue is just limited up to that of the areola. So this is just an image to, to show you how a breast cancer looks like. A unilateral breast carcinoma of a male, again a unilateral. I cannot emphasize more and more for this. The breast self examination, I think every young girl should also do that. In fact, now the standard for mammography is also changed. Like after 35 years of age, it is advisable to get a mammography done for every 6 months or 1 year. So you need to understand these steps and just look for these signs. This simple breast self examination can really help in the diagnosis as well as in the quick quick visit to a doctor where a diagnosis can be made. This
Thank
is
you, Dr.
just
Sneha. This was very informative. You still have some slides?
Ma'am, I've just put the slide for them to revise because I emphasize that the arterial supply is important. So they just need to know, and this is for the lymphatic drain. These 2 points you cannot forget.
So somebody has a question here. Somebody has posted a question why a man is required to take estrogen therapy. Did you mention something regarding estrogen therapy?
Ma'am, over here. Yes, ma'am. Ma'am, estrogen therapy, ma'am, that can be given in some kind of, for testicular, the feminizing syndromes and the testicular carcinomas. they will understand the testicular feminizing syndrome over here, and estrogen therapy can be given. And I just need to point one more thing. See, we have read about the axillary lymph nodes as, you know, anterior, posterior, lateral, but clinically the axillary lymph nodes are divided as level 1, 2 and 3. So the level 1 of the axillary lymph, it is in context with the pectoralis minor muscle. So the lymph nodes lying inferolateral to pectoralis minor are level 1, which are lying on the pectoralis minor are level 2, and which are lying superomedial to the pectoralis minor are level 3. So this is just for clinical and surgical relevance. The levels are divided 1, 2 and 3. And this is okay. You'll understand about all this later. And see. This is just a dissected specimen. I have taken it from Macman's, and over here you can see, you know, see this is how the ampulla, see the ampulla of the lactiferous ducts. I'll just follow these 1, 2 and 3. So first is the ampulla of the lactiferous ducts. Second is the condensed glandular tissue, see condensed. So this is the fatty part. These are the septae, and this is the condensed glandular tissue. Then 3 over here is the fascia, that is the pectoralis fascia which lying over the pectoralis major muscle. 4 is fat over here also, fat over here. 5th is the fibroseptums, that is the suspensory ligament of the Cooper. 6, these are the lactiferous ducts, nipple, 7th number. 8th is the pectoralis major muscle, okay, and 9th is the this potential space, over here, the small space is the retromammary space. And it is through this space that you are going to, you know, if you would have been dissecting in DH, you would have been separating the and have been provided with a female cadaver, you would be separating the breast, the breast from this level, because it is easily, it's a potential space, so you can just slide on and just separate the breast. And I think that's all. I'll just play the video. Thank you everyone, and just know when to call your doctor when you see these changes in you know, in the breast or in the nipples. Ma'am, should I play the videos now, the ones that we've recorded?
So they must have a look what the real specimen looks like.
Yes ma'am, I'll just is this visible to everyone? Just please reply. Just unmute yourself and just tell me. ठीक है ना? Visible है ना सबको ये? ठीक है. Okay.
Yes ma'am.
See, see, this is a female. See, this is a female cadaver. See, and we're just trying to show you. See, this is the sternum. So this is the parasternal area. We're just trying to tell you, you know, the margins of the breast, the medial extent, that is just lateral to the sternum. Then from here to here, the vertical extent is from the 2nd to 6th, but this is variable. With age, with pregnancy, it changes. With young females, and see, this is the lateral extent. Take care. That is the mid-axillary line. So this is the axilla, and that is the mid-axillary line. So since the cadaver is lying supine, so the breast being pendulous, they are just drooping on the sides, and they are not firm. See, so this is the superior extent. And then this is the inferior, and then this is the areola, and this is the nipple. Okay. Okay, I'll just play the second video. Okay. So this is a dissected specimen. Over here we are seeing the dissected specimen. We have removed it from the retromammary space. So see, this is the fat that you can see, the nipple, the areola, and these are the borders. So these are the fibroceptors that you can see from outside. So basically it is a glandular, fibrous and fatty tissue. See, this is the nipple. It's more clearly seen in this one. They are not from the same cadaver. So these two are, you know, from the different cadavers. So they're not from the same cadaver. See, pointed, conical, cylindrical, whatever can be the shape, then the area surrounding is rich in melanocytes. That is the areola. See, this is the axillary tail of Spence, which is projecting up to the anterior group of the axillary lymph nodes. See, this is the retromammary space, the smooth part, which lies over the pectoralis fascia muscle.
So the specimen can be removed entirely. It's entirely in the superficial fascia of the pectoral region and is separated from the pectoral fascia, which covers the pectoralis major muscle by loose areolar tissue. And that loose areolar tissue is actually it's lying in contact with the pectoral fascia with a little bit of loose areolar tissue. So there is no well-defined space there, but it is a potential space. It's not so much an actual space, but a potential space. Why it is defined is because clinically this space is very important. If there is any infiltrating carcinoma, this space is invaded first, and then the pectoral fascia and the pectoralis major is infiltrated, and that will cause the gland to be fixed to the underlying tissues. That is pectoral fascia, pectoralis major. So the retromammary space is a potential space with very little quantities of loose areolar tissue, and it's the reason why the breast is freely movable over the pectoral fascia. And this gland lies entirely in the superficial fascia. It's a modified sweat gland with a specific function. It is adapted for milk secretion, milk production and secretion and ejection, both. Okay, continue please.
Okay, ma'am. See over here, see, these are the, I'll just put it here, see. These are the, of course you cannot very clearly see right now, but these are the fibrous septae, that is the suspensory ligament of the Cooper, along with the lactiferous ducts. See, you cannot differentiate, of course, the septae and the ducts, they're almost looking the same, but this is how the architecture looks like, you know, when the lobes in the lobes are formed. And
You will have to piecemeal remove the entire fat to expose the breast parenchyma. So this is the intact mammary gland removed from the subcutaneous or superficial fascia.
Okay, so now this is a male cadaver. So we're just trying to show you the bed, that is the base of the breast. What we read, you know. So we'll just see. See, this is the skin. So you're going to reflect the skin. This is pectoralis major muscle. So this was forming the medial 2/3 of the base of the breast. The fibers of the pectoralis major muscle. See, the digitations of the serratus anterior. Like this, see. And then inferiorly we saw that inferior external oblique. External oblique was the muscle, so this is the external oblique. So pectoralis major, serratus anterior.
So here you can appreciate the posterior relations of the mammary gland. Even though this is a male cadaver, the relations will be common between male and female. So this is to show you all the muscles that the mammary gland may lie in contact with posteriorly. So this is the upper part of the external oblique, which is being shown. And the lower abdominal fibers of the pectoralis major take origin from this muscle.
So this was the rectus sheath. the external oblique aponeurosis and the rectus sheath. So we'll see all this. We'll see all.
Yeah, no, whatever it is, but the total ratio, I think that ratio we don't have to worry because there are templates now specified by MGI or specified by the institute or something like that. The idea is
Hello. Okay, so everybody is understood. We have seen the pictures also, and I think we're done. Ma'am, we are done with the lecture.
I'm sorry, for a minute the other meeting had started. Yes,
Ma'am, we're done.
I think it's very clear. Does anybody have any queries at all? Somebody is asking, are enzymes used to remove fat or manually? What does this mean? I'm not sure. You can open the chat box now and see if they have any queries. You can answer them.
Trying, ma'am. Otherwise, ma'am, they can post it in the group. I think that would be a better place. I'm understanding.
Absolutely. Because now I think it's nearly 5 o'clock.
Yes, ma'am. Ma'am, ma'am, the bhaiya is also waiting, ma'am.
The rest of the queries can be answered on the WhatsApp group. Fine.
Ma'am, change meeting.
And there's a faculty which I need to attend immediately. Okay? So fine. Please close the meeting.
Okay, ma'am. I hope everybody understood. Please, any questions, just text me in the WhatsApp group. Okay, thank you.
In the group, please, not individually.
Please in the group. In the group.
MBBS.
Yes, please.
Yeah.
Okay, thank you, everyone.
Thank you, Dr. Sneha.
Thank you, ma'am.
For a very informative lecture.
Thank you, ma'am.
We've all learned a lot. Thank you.
Thank you, ma'am. Thank you. भैया ये बस लीव मीटिंग कर दो ना एंड मीटिंग फॉर ऑल ओके एंड मीटिंग फॉर एवरीवन यस ठीक है हो गया भैया शट डाउन कर दो क्या आपको कुछ करना होगा इसमें आ जाओ फिर क्यों नहीं कर पाऊंगी क्या बात कर रहे हो आप मेरे को कंप्यूटर नहीं आता आप ऐसा कहना चाहते हो
नहीं आप करो एक बारी
ये लाइव स्ट्रीमिंग कैसे बंद होगा
एंड स्ट्रीम राइट